A very big hello to my wonderful children. Good afternoon. It's almost going to be evening. Let's see who all are here. So for those of you who are here, give me a quick hi. Let me know if you're able to hear me, if you're able to see me, if you're ready to score full in biology as well. Come on, children. I'll wait for a minute so that everyone else can join. We definitely know the timing is not that great. You guys might be just back from school, but still. Hi, Harshvardhan. A very good evening, dear. Okay, let's wait, Harsh, for a few more people to join, and then we will begin. Okay, so 4 people are here. Everyone is doing good? All of you ready for your midterm examination? Well, my dear children, if at all you're not ready, do not be worried about anything. You are going to absolutely ace the examination if you're giving 100%. Sunanda, very good evening, dear. Okay. I think then we'll begin. Right, 8 people are there. Manasa, yes, my dear children, a very good evening. M. Girish, definitely I remember you, and good evening ma'am, I'm the last batch student, I got 99 in biology. Thank you so much for coming and telling that. And Priyanka, hi dear. Okay, I see people out there. So shall we begin? Okay, so the first chapter that I have taken is Principles of Inheritance and Variation, and I'll be solving 28 questions. It includes the MCQs, it includes the 2-marker as well as the 5-marker questions, all the important ones. And I'm pretty sure if you are with me throughout the whole video, you are going to ace your biology examination and no one is going to stop you from that. Ready? Okay. Starting off with the first MCQ question that we have over here. Well, these are the type of questions that would be coming. 2-marker, long answer, and also what do you have? You would be having at least 1 MCQ. So according to one blueprint, 1 MCQ, second blueprint, 1 MCQ. Here also you see 1 MCQ has been coming. So definitely you can expect something like that over here. And now, just give me a second. Yes, the first question that we have. So what's the policy? I read out the question and you tell me the answer before I do. So 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color, normal body, and white eye color miniature body is due to. Straight, I'll ask you the straight question. That is, they are telling you that in Drosophila 37.2% recombination is there. What do you understand from that? Any guesses, my dear children? Any of you guys? I'll give you a very simple theory, okay? That is, if 2 genes are very close to each other, their recombination rate would be very less. And what do you call these genes as which are very close to each other? You call them as tightly linked genes. And how do you remember it? You have a best friend and you think he's your best friend, okay? You would not allow any of your other friends to come in your gang. Why? Because you are very much close. So there is no chance for a new gang formation. There is no chance for a recombination to happen. So over here, tightly linked genes would show lesser recombination. On the other hand, if the genes are very far away from each other, my dear children, they have too much distance. You call this as loosely linked genes. Okay, and they will be having more amount of recombination. That means you and one of your normal friend. Obviously, you can include one more person. There is chances for what? More recombination to happen in that particular case. And we will say the recombination rate is high. So over here, 37.2% recombination. Is it too much recombination or too less recombination? It is actually too much recombination. So what do we understand? That this criteria is being followed. That means they are loosely linked. And when they are loosely linked, are they near or far from each other? They are going to be far. And now you tell me the answer. Hi Nagaraj. Since I've given you all the clues, I want the answer from you guys. If someone is showing 37.2% recombination, I have told you it would be loosely linked. And if they are loosely linked, will they have more distance between them or less distance between them? Come on children, Harshvardhan says it's D. D is tightly linked, no? Can it be tightly linked by any chance? 37.2% recombination is very much recombination. And if that is happening, it has to be, okay. C. Loosely linked and longer distance between genes. Exactly. No problem, Harsh. It's all cool. Yeah, he has given me the correct answer. So Sunanda, Nagaraj, Harshvardhan, all of you saying it's C. So I'll lock C and your answer is actually C. Always remember 50% recombination is the highest recombination ever possible. So if at all they're saying 37.2, that means recombination is there. If at all new combination is there, that means you and your friend are very far from each other. There are chances someone else can also get in. So, okay. Over here, tightly linked genes will show lesser recombination. For example, I say 1.7% recombination. That means they are tightly linked. Cool. Let's go ahead with the next question out there. Kudos to everyone who got it correct. Before I go ahead with the next question, I want to give you a very important information. Have you heard about KC90? If at all not, then hear me out carefully right now. Because my dear children, it's a course that we are going to launch really soon and the registration has already opened. This is aimed for KCET as well as for Comet K preparation. So it's not just about your 12th. After 12th, you would be doing something. And suppose you're going to go into engineering or BSc nursing or agricultural fields, you would have to crack KCET examination for getting into a very good college out there. And if at all you're planning to prepare for KCET, then this course is perfect for you. We guarantee that if at all you work hard with us, you are going to get a rank within 1,000. So if you have any queries regarding that, you can call this number or also go to the website directly. Well, it is offline. As of now, we considered it to be offline. It would be happening at Diksha campuses. But if at all you think that you need it online as well, you can let us know. We might change our minds. Okay? Ready for the next question? Chalo. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 ratio. It denotes that. What does it denote? 9:3:3:1. So those of you tell me this answer, I will give you chocolate if I meet you in person. Which I do not know if it's going to happen here or now or not. So 9:3:3:1 ratio means he actually took two type of plants. One had round and yellow seeds, okay, and the other plant had wrinkled and green seeds. Now when he crossed, obviously you will expect either this to come or this to come. You don't expect them something new to come up there. But over here something new came up. So 9 of them were this, the dominant character, that was round and yellow. One was the recessive character, which was what? Wrinkled and green. And you know what about the term? Both of them, they were new. So here round was, instead of being with yellow, it was with green. So they found round and green seeds. And here wrinkled, which was usually with what? Green, now it was yellow. So what do you see? You see new combination. Why do you think new combinations are up there? Nagaraj says D. The alleles of two genes are segregating independently. Is that so? Well, let's see. So over here, my dear children, alleles for round and yellow looks like this. Okay. Suppose it's like this. Capital Y, small y. Suppose it was round and yellow like this. Capital R dominant over this, which is why it is round, and capital Y dominant over this, which is why it is yellow. Okay. So now over here, when you form the gametes, from this pair, one guy can come in. So from this pair, capital R can come in. From this pair, one guy can come in, it can be capital Y. Similarly, capital R can come in from here and small y can come in from here. Similarly, small r can come in from here, capital Y can come in from this group. And small r and small y can also come. So these are the gametes, new different type of gametes. Why do you get different type of gametes? Because alleles, these are alleles of one gene, that is the the structure, and these are the alleles of another gene which codes the color. So both of them are going to segregate independently. Capital R does not say I will go only with capital Y. It is not like that. They are segregating independently, and since they are doing that, which is why we are going to get what? Different things out there. Otherwise we would not be getting 9:3:3:1 ratio. So you are absolutely correct, Nagraj as well as Mansa. The answer is what? It's D. Kudos. So those of you who are getting your answers correct, my dear children, kudos to you. Those of you are getting your answers wrong, do not be demotivated. Write it down somewhere. This question, I read it wrong, and attempt it later on after the video ends. Done? Deal? Chalo. Next question. The pedigree chart is given below. Identify the trait responsible for this inheritance pattern. Can you try it out? This is the pedigree chart. Okay, over here the parents are not affected, because if they are affected it would have been colored. Here they are not affected, but the children, if you see, one boy and one girl is affected. Now these two children, they made it, and now you have another child who is getting affected in the next generation. So what do you understand from that? What type of pedigree is this? First of all, you have to understand whether it is what do you call dominant or recessive. How do you understand that? If at all the trait or the disease is skipping generation, then it is recessive disorder. For example, suppose here also someone was diseased, here also someone is diseased, in the next generation also someone is diseased, you will say it is dominant disorder. But since here the parents are not diseased, okay, and the children are, here also the parents are not diseased but the child is. So what do you get to understand from this? That it is what? A recessive disorder. Now you have to understand whether it's sex linked or it is autosomal. That is the next query. So how do we need to find out? So before that, I've got certain answers. Nagaraj says it's D. Okay. Mansa says it's D. Nagaraj says it's B. What do you think? It's the answer for this question. Over here the male is affected. Here the female is affected. Okay, and here the male is affected. But it's not like the females are not affected or basically it's just the male who is getting affected. If at all it was like that, okay, if at all it was like that, you would have said that males, it is what? Sex-linked recessive disorder. But since here, we get a general idea that male and female, both of them are affected. So it's not here, but still it is happening up over here, right? So you will say that it is an autosomal recessive disorder. This is not sex-linked recessive disorder. Your answer would be B. Yes, Harshvardhan and Nagaraj got it correct. Okay, kudos to you guys. And Mansa, very close. But I get it. Where does the confusion come from? Because we see boy is affected here, boy is affected here. It's not like that. When it is X-linked, the parents from the male parent, it will be coming here. So male parent will be affected and the male child will also be affected. You can clearly see that. Okay? Fine. Let's go to the next question. So your answer would be autosomal recessive trait. Next question. Which of the following is not a recessive gene-linked disorder? Now this answer only those children would be getting who knows the NCERT properly. It is asked which is not a recessive gene-linked disorder. Sickle cell anemia is X-linked autosomal recessive disorder. Hemophilia is X-linked recessive disorder. Color blindness is X-linked recessive disorder. In your NCERT, you mostly, when you study about things, it is mostly about recessive disorders. Okay, some is X-linked, some is autosomal. So now, Nagaraj as well as Harshvardhan, both of them are saying A. Poma is here. Hi Poma, you're also saying A, and I'll say you all are correct and you all are good children because you're reading NCERT very, very well. So in the picture given, okay, of the pedigree chart, they have given an example of myotonic dystrophy, which is what? An autosomal dominant disorder. So this is autosomal dominant. So this one is the odd one out. Kudos to you guys. You guys are absolutely awesome. And here is the picture that I was talking about. So this is the autosomal dominant recessive disorder, which is myotonic dystrophy. In page number 73 of your NCERT, it is given. Cool? Chalo. Next question. A plant having the genotype capital A small a, capital B small b will produce dash kinds of gametes. Come on. How many type of gametes will it produce? Capital A, small a, capital B, small b. There's a small formula that you use for finding the answer for this question. Any guesses? What is the formula? And dear children, if at all you have chosen biology stream, if at all you have your classmates who do not understand biology better, or if you feel the biology is boring, send it to them as well. Okay? So question 5, Nagaraj says is B. Too much competition out there. Very much apt. Let's see if he's correct or not. So he says that if parents are like this, it will produce 4 type of gametes. How? Ma'am, I do not understand. Any idea? Can I say? So there is a formula that is 2 raised to the power n. N means the number of heterozygotes. So capital A, small a is heterozygous condition. If it was capital A, it was homozygous condition. So this is one heterozygote. Capital B, small b, this is another heterozygote. So in total, how many heterozygotes do we have? We have 2. So n is equals to what? 2. So 2 raised to the power 2 is equals to what? 4. So this would be the number of gametes. Got it? Okay, so your answer would be B. So kudos to you, Nagaraj, as well as Harshvardhan. And I'm not able to read your name. Can you please write it? Okay. So yes, all of you doing absolutely great. Your answer is B. Please do not forget this formula. I included this question thinking that it might come. Okay. Next question. It is, okay. Which of the following is not a pair of opposing characteristics Mendel studied? Which of them is not something that Mendel actually studied? Any guesses? Axial and terminal flowers, full and constricted pods, green and yellow pods, pink and white flowers. I suppose it was something else. It was not pink and white flowers. Any guesses? Harshvardhan says B. Check, check, check. I'm asking about question number 6. Are we answering question number 6 itself? Question number 6. Answer is Trishankar Dombali. Okay, Dombali, you are absolutely right when you said the answer, and I'm very happy to see you over here. So question number 6, the answer is pink and white flowers. In pea plants, it is violet and white flowers. The violet ones are dominant and the white one is the recessive character. Okay, so your answer would be what? D. What is the answer? D. So this is the list of characters. Okay, always remember the pea that we eat is in green in color. Right, it is green in color, but actually yellow is the one which is dominant. Yellow seed is dominant. Now if you talk about the pod, inside the pod you have the green peas, right? So the pod which you take, do you pick yellow ones or the green ones? It is a green one, so green one is the dominant one. Now height, tall is the dominant and dwarf is recessive. Okay, now pod, will you take something that is dry, that is constricted? No, you will take something that is full, right? So that is full is what we call full is what? Dominant character. So this is how you need to remember. So there are certain things over here and you need to remember like that. Okay, so the odd one out is D. Yes, I got you. Trishankar, I'll never forget your name. Okay, question number 7. Chalo. In the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are. This is dihybrid cross experiment. In that you are asked, in the F2 generation, okay, when you are crossing capital R small r, capital Y small I, round and yellow with another round and yellow, what is the number of phenotypes that you get? What are the number of genotypes that you get? Any guesses? Okay, D, A. Children, can I write the number of question as well so that I get to understand which question are you answering? Harshvardhan, is this 7th question that we are answering? Check, check, check. Remember, number of phenotypes means how many physical appearances do you see? Round and yellow is one, round and green is one, wrinkled and yellow is one, wrinkled and green is one. So 4 type of things I can clearly see. So 4 phenotypes are definitely there. What about the genotypes? And when genotypes means genetic makeup. That means this is round and yellow, this again is round and yellow. Okay, you get it? So you have to let me know like that. Okay, this is round and yellow, this is also round and yellow. So this is another genotype, this is another genotype. Fine. 7th D. 7th D, who said that? Poma said that it is 7th D and you are absolutely correct. It is this. So this is a question where you get confused, which is why I have put it over here. So number of genotypes, if you see, they are 9. They are not 16. Always remember, it is not 16. 16 means 1, 2, 3, 5. You are counting all of them. But some of them are getting repeated. So you do not have to count the repeated numbers. So there are 9 unique genotypes. Okay. So 4 and 9 are the things that you need to remember. Clear? So Harshvardhan says D. Nagaraj also said D. Okay. So Shankar, we are close. Okay, going ahead with the next question. Mendel's work was rediscovered by 3 scientists in the year. In which year was Mendel's work rediscovered? You know, Mendel was thought that it was not a good idea for him to perform the experiment, this and that. He used mathematics. There were so many things that happened. But 3 scientists came up in one particular year and said that, no, Mendel was actually correct with his experiments. Their name was De Vries, Correns and Schemark. Please remember, De Vries, Correns and Schemark. This question can also be asked. Okay, so Nagaraj is saying 8B. Does everyone agree with that? Question number 8, the answer is B. Yes, it is 1900. Always remember, it's 1900 when the 3 scientists, De Vries, Correns and Schemark, came and said that, yeah, Mendel was correct and his work were rediscovered. Okay. So 1900, it's not 1910, nothing like that. Cool, cool, cool. Next question. Genes located very close to one another on the same chromosomes tend to be transmitted together and are called as. Now this question only those children would be able to answer who heard me very nicely when I was explaining. Harshvardhan, you were correct dear. Okay, 8th answer is B only. So now we are in the 9th question, and any guesses? The genes which are very close to each other And when this chromosome is going to go, the genes also going to go with it. Okay, so they are going to be transmitted together. Both of them will be here itself, close to each other. What do you call them? Do you have any idea? C. Okay. Nagaraj is asking, is question number 9's answer C? Well, you are correct. It is linked genes. Both of them, they are tightly linked to each other. So you call them linked genes. Poma, you're absolutely correct, my dear children. Sorry. So your answer is C. Okay. So see, this is how whenever you study a concept and whatever questions comes around it, you will be able to answer it properly if you understand the concepts well. Okay. So your answer is C. You do not have to go with any other options out there. Chalo. Next question, chromosome maps or genetic maps were prepared by, what do you mean by that? Genetic map or chromosome maps. So over here I am saying basically this and these genes which are there, they are far away from each other and they show this much amount of recombination, that is 37.2% or whatever is there. Okay, and when I got to know that this much percent recombination is there, I also understood that they are far from each other. So I was able to locate A is over here, B is over here. This is known as genetic map or chromosomal map. Who was the one who discovered it? Nagaraj says option number D. Before that, Poma said 10th D. You are absolutely correct. The answer is Sturtevant. So you know what? The question is asked, who discovered linkage and recombination and everything? It was Thomas Hunt Morgan. Now, Thomas Hunt Morgan discovered all of that, but his student Sturtevant was the one who gave the gene maps. Okay, based on the information that was given by Morgan, he used it and came up with a concept that is known as gene mapping. So your answer would be D. His full name is Alfred Sturtevant. Okay, similarly, can I expect you guys also, after studying from me, can I expect you guys also would be telling the correct answers or probably framing new nice answers on the day of examination as well? If at all yes, then let me know here by clicking on the like button, and also you can put it down, yes ma'am, no ma'am, whatever you feel so. Listen, I am here for you to ask me question as well as for us to talk properly, so that if at all there is any doubt, it can be cleared up. So if at all you sit quietly and only give me the answers, it will be very boring class. So please talk to me and definitely yes, pay attention. Question number 11. A couple has 3 daughters. What is the possibility of them having a son next time? Yes, Nagaraj is in. So a couple had 3 daughters. What is the possibility of them having a son next time? What do you think so? Poma says 11th answer is C. 50%. How do you say so? Are you sure? Well, my dear children, a couple, that means a father XY and a mother XX, right? They had 3 daughters, okay. But the next time over here, he can contribute X chromosome or Y chromosome. Similarly here, X chromosome or X chromosome. This and this comes, suppose this gamete and this gamete fuses, you get XX, it's a female. This and this fuses, again you get XX, again a female. This and this fuses XY, that is a male. This and this XY, that is a male. Now you tell me, 50% chance of the male to be formed and 50% chance is there for the female to be formed. So the next child can be what? A male or a female. So for a son to be formed, it is what? 50% chance. So you are absolutely correct, Poma. And Nagaraj, correct. Okay, she said she was sure and I could see that. Very much nice. Okay, let's go ahead with the next question. Ayyo, where is it? Okay. When is it possible for a woman to be colorblind? Harshvardhan also said C. Kudos. Kudos, Harshvardhan, you're doing great. Absolutely great. So when is it possible for a woman to be colorblind? Any idea? The father has a normal vision and the mother is a carrier. So color blindness is an autosomal recessive disorder. What do you understand from that? Autosomal recessive disorder means someone who has both the chromosomes to be, it is an X-linked, color blindness is a sex-linked recessive, it is not autosomal, it is sex-linked recessive disorder. So over here, both the X chromosomes which are there, which are going to come together, both should be affected, or if at all it's a male, at least one should be affected. Okay, at least one should be affected. In that condition only, my dear children, what will be happening? You will get color blindness. Okay, so ma'am, if the condition is like this, one X chromosome is affected, then no, the other one will overshadow and this particular female will be just a carrier. Now you are asked, when can a woman be colorblind? That means she should be getting one chromosome from her father and one chromosome from her mother, right? So in this condition, her father should be definitely colorblind. because he has to contribute this. Her mother can be what? A carrier, my dear children. Can be a carrier, right? So over here, suppose this and this comes together. You get a female who has what? Color blindness. On the other hand, there can be one more condition where what is happening? The father is colorblind and the mother is also colorblind. In this particular condition also, the women can be colorblind. Why? Because this and this comes together, you get anyway XC. So now let's see. The father has normal vision, that is not possible. The father has to be colorblind. So these two options goes crossed. The father is colorblind and the mother has a normal vision. Okay, the father is colorblind and the mother is carrier. When you say that, it becomes more specific because normal, this is also normal vision. She would be having normal vision, she would be a carrier. So if you just say normal vision, it can be just like this also. So to avoid that confusion, I think this answer would be the best one. So mother is a carrier. Okay, you have to at least say that. So your answer would be what? D, my dear children. Yes, Trishank, very good. Nagaraj, Harshvardhan, all of you, Poma, absolutely great. Okay, so this is your answer. I hope you guys are understanding. Yes. Now, a man marries a woman and both do not show any apparent traits of inherited disease. Listen, this particular question is very important and very nice also. You will have fun doing this. A man marries a woman, okay, and both do not show any apparent traits of inherited disease. Both are not showing any diseases traits. 5 sons and 2 daughters are born. 3 of their sons suffer from a disease. However, none of the daughters are affected. Daughters are not affected. The following mode of inheritance for the disease is 13A, sex-linked recessive. Is it sex-linked recessive? 13A, Poma says 13A. So both of them should not be showing any. Suppose this is the thing. It can be a condition like this, right? The male is fine and the female is like this. Suppose now this and this comes together, it is XCY, the male will be affected. But the female, my dear children, whatever you do, over here, the father has just a plain X chromosome. She will be definitely a carrier. She will not show the disease. So yes, this satisfies the thing. This can be what? A sex-linked recessive disorder. So your answer would be this. Okay, the answer is A. Nagaraj said A. Poma, before that she said A. Correct. Okay, so your answer is sex-linked recessive disorder. So whenever you say that 3 of their sons, out of 5, 3 are them affected, you get to know that males are getting affected more, so it has to be sex-linked. And now you get to know that it's recessive also. Okay? Fine. Chalo, next question. If both parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? Both of them are unaffected carriers. That means they have one gene which is which has mutation, but the other gene is normal. Okay, it is an autosomal recessive disorder. So over here, capital R, small r is taken. Similarly, over here, capital R, small r is taken. You are asked, what are the chances of pregnancy resulting in an affected child? You are not asked about carriers. So to being affected, my dear children, capital R, small r, capital R, small r. What happens? Over here, it can be capital R or small r, capital R or small r. This and this goes, you get capital R, this and this goes, capital R, small r. No, none of them are affected because you need recessive condition. This and this again not affected, this and this affected. So out of 4, only 1. So 1 out of 4 means what? 25%, my dear children, will be what? Affected. So 14th answer would be A. Yes, Poma, yes, Nagraj, yes, Trishank, Harshvardhan, all of you correct. Doing great. Okay, absolutely great. So your answer is 25%. Next question. The fact that every gamete formed has either of the alleles for a trait follows what? The fact that whatever gamete is being formed has either of the alleles. That means, for example, I take X chromosome over here, Y chromosome over here. So I am saying that the gametes which are formed, it will either have X or it will either have Y. What law does it follow? It's a very easy question, but children sometimes get panicked. See, how can an easy question come up there? So we have a new person out there, Sonu. Hi, dear Sonu. So Sonu says it's D. Is it D? Every gamete formed has either of the alleles for a trait. Think more. Nagaraj says it's C. Law of segregation. Yes, my dear children. What does law of segregation says? That whenever gametes are formed from a pair of from a pair of alleles, only one would be going. So that is what you mean. They are going to separate and only one could go, either this or this. Okay, either this or this. That means whatever alleles are over here, suppose A allele is over here, capital A, small a allele is over here. So here you will have capital A, here you will have small a. Both of them are separating and going. This is known as law of segregation. Got it? So 15th answer is C. Why there's a confusion? Concept of unit factors are. No. Okay, so those of you who answered wrong, I hope it's clear. If it's clear, can you give me a quick thumbs up? You all know where did you get wrong, right? It's law of segregation. If law of independent assortment, I'll tell you when it's applied. Suppose you're talking about two things, okay, two type of alleles, capital A, small a, and then capital B, small b. Over here, this is one set of allele, this is another set of allele. When both of them are going to segregate, they are going to segregate independently. This chromosome can come and this chromosome can come in one gamete. This chromosome can come, this chromosome can come in one gamete. That is independent assortment. Over here, only one. Okay, so yes, Poma said yes, I get your yes, so I'll go forward, and kudos to all of you who are trying to answer. Now, my dear children, the 1 marker questions, I mean the most repeated type of questions I have completed, or the most unique ones I have completed, and now we are going ahead with the 5 marker questions first, and then we'll go to the 2 marker. So what is phenylketonuria? Write the cause and the symptoms of phenylketonuria. I have given you the whole thing over here. You can copy it just for making you understand. I'll tell you what is phenylketonuria. First of all, it is autosomal recessive disorder. In your book, apart from color blindness and hemophilia, everything else is autosomal recessive disorder. Color blindness and hemophilia, sex-linked recessive disorder. And one dominant disorder is given, and that was what? Myotonic dystrophy. So phenylketonuria being autosomal recessive disorder, over here what is being happening is that one particular enzyme which is known as phenylalanine hydroxylase, this particular enzyme is not being produced. Okay, and when it is not being produced, what happens, my dear children? Phenylalanine, which is what? Phenylalanine. I think I have written somewhere, wait just a second. Yeah, phenylalanine, which is an amino acid, it is not able to convert to what? Tyrosine. It would not be converting to tyrosine, which is another amino acid. And when it is not getting converted, what will happen? It is going to accumulate over here in the form of phenylpyruvic acid. And when it's accumulated, my dear children, what happens in our body? We are going to get mental retardation. We are going to have mental disabilities happening up there. Apart from that, since these things cannot be reabsorbed by the body, it is excreted in the urine. Okay, it is excreted through the urine, which is why the urine smells a little bad. It has a foul smell because it cannot be retaken by the kidneys as well. It cannot be reabsorbed in the kidneys. Fine. And one more very important thing, tyrosine is not being formed. Okay, since tyrosine is not being formed, melanin is not going to be formed. Melanin is something that gives us skin pigmentation, hair pigmentation, everything. And now since melanin is not being formed, the people who have this will also have reduced pigmentation in their skin and their hair. Okay, done. Okay. Ma'am, where can I get the PDF of this class? I'm so sorry. I'll be giving you the PDF once the class ends. Okay, definitely will be giving you, and I'll ask Aishwarya ma'am to also give an update about yesterday's chemistry class. Done, Pama and Harshvardhan? Okay, so over here, my dear children, these are the 2 things that you need to remember, and I have clearly put it down over here. Phenylketonuria is also an example of what? Also an example of pleiotropy. Pleiotropy means what? One gene. If one gene is mutated, it can have multiple effects. So as you have seen over here, one gene is getting affected and it has multiple effects. One is mental retardation, one is what? Reduced pigmentation. I hope that is clear. So you have to write all of this thing, write the answer properly, what do you mean by that, then show the symptoms. Okay, what exactly happens over here, and then tell what are the symptoms of this particular disease. That's all. Fine. Now can we go ahead with the next question? So the next 5 marker question that you can get, T.H. Morgan selected fruit flies for his genetical experiments. Why? Do you have any idea why did he pick fruit flies, nothing else? Out there, does anyone know the scientific name for fruit flies? Anyone? Scientific name for fruit flies? Can you let me know? Because I don't know. Come on. Now let's have a talk also in between. Otherwise, I'm pretty sure it gets boring. What is the scientific name for fruit flies on which Thomas Hunt Morgan was working? No one? Ayyo. Drosophila melanogaster. Exactly, Harshvardhan. It is known as Drosophila melanogaster. Okay, and the features because of which he actually took them is that it can be grown on a very simple synthetic media. You do not need hi-fi things out there. They complete their life cycle within just 2 weeks. So for example, I want to see if at all there are any changes. I just have to wait for 2 weeks. That's all. Single mating could produce a large number of progeny, so I do not have to do multiple matings out there. Single mating can produce what? Enough number of progenies. There was clear differentiation of sexes, that is females look different from males. You know what? Females were larger in size than the males. If at all the picture is given of Drosophila, you should know. So the smaller one is the male and the larger one is the female. So these are the few characteristics that you can write out there. Nagaraj, yes my dear children, it's Drosophila melanogaster. Now there's one more question. What are chromosomal disorder? Give an example. Chromosomal disorder means what? Mendelian disorder means what? So over here, suppose this is a chromosome and there is a gene. This gene gets mutated, which is why whatever protein had to be made was just not making, which is why you get a disease. This is known as Mendelian disorder. Now suppose this whole chromosome itself didn't come into a cell, my dear children. It got lost somewhere. Okay, or suppose instead of having one chromosome, I have one more extra chromosome. So these type of things lead to chromosomal disorder. Example, Down syndrome. Example, Turner syndrome. Example, Klinefelter syndrome. These are the three examples of chromosomal disorders. Clear? If you're asked about Mendelian disorders, you have to write about all those things, color blindness, this and that, because all of that, the genes are being mutated on the X chromosome, this chromosome, that chromosome. Works? Cool. I hope you are understanding this. Now going ahead to the next question. Few autosome-linked recessive gene blood disorders occur in human population. Among them, some are related to qualitative and quantitative problems of synthesizing blood proteins. Can you tell me what is the blood disorder which is quantitative and which is the one which is qualitative? I'm talking about Mendelian disorders. There are two diseases, my dear children. Okay. Both of them are blood disorders. It causes trouble in the blood itself. But one is quantitative and the other one is qualitative. Any idea? Any guesses? Fast, fast, fast. Oh my God, you can clearly see it on the screen as well now. So this one is thalassemia. Okay, and it is what? It is a quantitative disorder. Why do I call it quantitative disorder? Ma'am, is there more MCQs on other chapters? Yes. In every chapter that we do, we'll start off with MCQs only. Okay. So over here, I am planning to cover your MCQ, your most probable 5 marker, your 3 marker, whatever questions come from the chapter, and then we will move on. Okay. So that your whole marks is safe out there. You get those marks. So thalassemia is quantitative. Yes. Why do you call it quantitative, my dear children? Because over here, I suppose you know that hemoglobin, when you talk about hemoglobin that carries oxygen, my dear children, hemoglobin is made up of two types of polypeptides. One is alpha and the other one is beta. Okay, alpha globin chain and beta globin chain. So alpha globin chain is coded by two genes that you see over here. This is one set of gene, this is another set of gene. One gene has 2 alleles, another gene also has 2 alleles. Suppose over here there is some mutation in this particular allele. What will happen? Alpha globin chain is going to be produced, but not too much. Suppose both of them are mutated. Still, alpha globin chain is going to be produced with the help of these alleles, but not too much. Suppose this is also gone. Then alpha globin chain is not going to be produced at all. So this is how you say you have probably thalassemia. Okay, so thalassemia minor patients have a few, do not have the other ones. Thalassemia major patients are the one that do not have it. It's very risky. They do not have alpha globin in their body. You get it? Same thing goes for beta thalassemia. In beta thalassemia, the beta globin chain is not being produced. Okay, so it can be either, suppose this is not there, this is mutated. So still this one is there and it can be produced. Suppose both of them are mutated, then beta globin chain is not going to be produced. So this is quantitative. The quantity is getting reduced or totally reduced and things like that. On the other hand, as Nagaraj is saying right there, sickle cell anemia is a qualitative disorder. Why qualitative? Because over here beta globin chain, it is getting produced, but the quality of it is very bad. So what exactly happens is that all the globin chains, it's a protein, okay, beta globin, alpha globin, everything is a protein, and proteins are made up of amino acids, okay, and who gives the information to form amino acid? It's the mRNA that gives the information. So over here the codon was GAG for a particular amino acid that is known as glutamic acid. Now over here there is a mutation that is happening. A, which is there, it is replaced by U. So which is why instead of having GAG, which was coding for glutamine or glutamic acid, over here it is glutamic acid, okay, and over here it is going to produce valine. And one amino acid, if it's changed, my dear children, the whole protein structure would obviously change, which is why we have a different beta globin chain or some mutation happening in the beta globin chain being produced. Now if beta globin chain is produced, beta globin chain is mutated, the hemoglobin which is made up out of it, it also gets destroyed, my dear children, somehow. It won't be able to carry enough amount of oxygen. If it is not able to carry enough amount of oxygen, the RBCs which carries this hemoglobin, they get into sickle shape. Okay, what happens to them? They become sickle in shape. So that is the thing about sickle cell anemia. And how do you need to write it? You need to write it like this. First of all, you have to mention about sickle cell anemia, say that it's a qualitative defect. What exactly happens over here? There is a mutation in HBB gene, hemoglobin beta gene, that's what you mean. So instead of glutamic acid being produced, valine is being produced. At what? At the 6th position. So this is an mRNA. In mRNA you have codons 1, 2, 3, 4, 5, 6, like that. So this is one codon, this is second codon, third codon. On the 6th position this is happening. It was GUG. Okay, sorry, it was, just a second, it was GAG. And then it is getting replaced by GUG. Always remember A comes before U. So A was there first. Now this was coding for what? Glutamic acid. This is one amino acid. Okay, but it was replaced by what? Valine now. So G comes before V. So you can remember glutamic acid was there and it is replaced by valine later on. Okay, got it? So that is a substitution that is happening. Here it produces abnormal HBS. That is abnormal hemoglobin instead of HbA. That is a normal one. So HBS homozygous disease, that means if both the, suppose over here on both this chromosome, this is getting affected and this is also affected. So I'll say HBS and HBS. This homozygous condition is where you get sickle cell anemia. Okay, suppose it is like this. One is HbA, that is a normal one, and the other one is HBS, then I'll say that this is a carrier of sickle cell anemia. This person would not show the disease. Suppose it is HbA and HbA, then this person is totally normal. Okay, so this is the thing that you need to remember. Now, so please remember homozygous condition, what happens, heterozygous condition, what happens. Now in thalassemia, it is a quantitative defect. It can be caused a mutation in the HbA1 or HbA2 gene, that is for alpha chain, and HbB for beta chain. Okay, where do you find this? On chromosome number 16. Where do you find this? On chromosome number 11. So B and B looks the same, you can say 1, 1. Okay, so chromosome number 11 and here it is chromosome number 16. Hemoglobin formed is normal in quality but reduced in what? Quantity. Leads to anemia, weakness and fatigue. Okay, because obviously you do not get anything out there. Oxygen is not there, so you will get fatigue. Hemoglobin is not being produced, so obviously anemia will be there. Okay, now severity depends on whether one or both the alleles are defective, as I told you. Okay, so here suppose only this is affected but the other ones are normal, it's totally fine. So severity depends on that. I hope this is clear. As simple as that. Now I'm asking you one question right now and you have to tell me the answer for that. There is another blood disorder mentioned in your NCERT which was in the lineage of Queen Victoria. Which is that blood disorder? It is not a autosomal recessive disorder. I have given you a clue. It is not an autosomal recessive disorder. What is that blood disorder? It is what? Come on, come on, come on. So children who are paying attention to this, who are listening to me carefully, will be able to tell the answer for this question. Another blood disorder, apart from thalassemia, apart from sickle cell anemia, one more blood disorder is there. Any guesses? What is that? No one? Come on, come on, come on. Children? I think everyone went to open the NCERT and see the answer. Okay, since you guys are not saying, I'll say. It is hemophilia. It's a sex-linked recessive disorder, which is also a blood disorder where what happens? A clotting enzyme, okay, a clotting enzyme is not being formed. Clotting enzyme is something that helps in the clot. For example, I get a cut. My clotting factors work really well, which is why the blood gets clotted and the blood does not go out. But suppose that particular factor is not being produced, do you think clotting will happen? No. Okay, so that is example of a blood disorder. It is hemophilia. It is sex-linked recessive disorder. Yes, Nagaraj. So simple cut results in non-stop bleeding in hemophilia affected individuals. Why? Give reasons for the following. Why do you think it results in this? Because, my dear children, what happens? A single protein that is a part of the cascade of protein involved in the clotting of the blood is affected. The gene that codes for this particular clotting factor is getting affected, which is why clotting does not happen. Okay, so this is the answer for this question. Now the next question is what? Turner syndrome affected females are usually sterile. That means they cannot have babies. They are sterile in nature. Why? Because over here in Turner syndrome, instead of having XX chromosome, they have lost one chromosome. This is chromosomal disorder. So they just have X chromosome, and you say this condition as XO condition. So which is why, since they do not have 2X chromosome, what will happen, my dear children? They will be having rudimentary ovaries. That means their ovaries are not well developed, which is why they would be sterile in nature. Got it? Works. So the same goes the answer like this. Females are usually sterile because they have only one X chromosome, leading to underdeveloped ovaries and lack of what? Normal gamete production. Cool. Now next one. In Morgan's dihabit cross experiment on Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. I told you, if at all you remember the funda, less recombination means what? Less recombination means what? They are very tightly linked. So you have to explain that. That less recombination is there because why? The genes coding for all this thing which is being written over here, it is what? Tightly linked. They have very less distance between them. They are very close to each other. So understand a simple funda and you will be able to answer everything. So yellow body and white eye genes in Drosophila show less recombination because they are located close to each other on the same chromosome. Exhibit what? Exhibiting genetic linkage. Are you getting it? Or is it me? Is it me, Jasmine ma'am, who is just talking? Children, how do you let me know if you're getting it or not? Click on the heart button. Click on the like button. Put like this. Then only I'll get to understand whether you are listening to me or not. Okay? Yes, Poma. Thank you so much. Fine. Skin color. Now next question is what? Inheritance of skin color in humans show different phenotypes. What do you mean by that? Basically, you and me, do we have similar skin color? No. Why? Because it is showing polygenic inheritance. Remember, it is example of polygenic inheritance. What do you mean by that, ma'am? So usually we have studied, Mendel said that one gene is there that is coding for one particular character. Capital T for tallness, small t for dwarfness. But does it usually work like that? In our case, no. So there is a character that is skin color which is dependent on so many genes. Multiple genes are affecting it, your skin color. So it can be, suppose over here capital A, capital B, and capital C is there. What is the genotype of this particular human being? It is capital A, capital B, capital C, capital C. That means this person has the darkest skin color. They would be having what? Darkest skin color. On the other hand, suppose I am shifting this capital A and making it small a. What will happen? Now the genotype is capital A, small a, capital B, capital C, capital C. So over here the skin color has reduced a bit. What do you understand from that? That means multiple genes are coding for one particular trait. They are all together responsible for one trait. Suppose here it is small b, again the skin color goes lighter. Suppose here it is small c, again the skin color goes lighter. It is small a, small b, small c. This will be the lightest skin tone. So since multiple genes are coding for a character, you call this as polygenic inheritance. The same goes with your height as well. Multiple genes are responsible for that. So skin color in humans, it shows different phenotypes due to polygenic inheritance, where multiple genes contribute to the phenotype, leading to what? Range of skin color variation. That is why we have different things. Okay. Yes, Nagaraj. Yes. I'm really happy when you guys tell me the answers like this. Great going. Now one more question. Accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why do you think phenylalanine is getting accumulated? There is no one to convert phenylalanine to tyrosine. Because in phenylketonuria, the gene which codes for phenylalanine hydroxylase is affected. So you have to tell about that. Okay, so phenylalanine accumulates in the body due to defect in the enzyme phenylalanine hydroxylase, which is the one that converts it. That is why it is getting accumulated. There is no one to convert it. Simple. Okay. Now, 20th question. Define aneuploidy. Give 2 examples of aneuploidy. Mention the karyotype of Klinefelter syndrome of an individual. Okay. So what do you mean by aneuploidy? Any guesses? Anyone out there? Aneuploidy basically means, suppose I needed to have 23 chromosomes, but I have one more extra chromosome. This is an example of aneuploidy. I needed to have 23 chromosomes, but I just have 22 of them. One is lost. Okay, 23 minus 1, 1 is lost. This is also a case of what? Aneuploidy, my dear children, where basically what is happening? A proper segregation is not happening. We say non-disjunction. Okay, failure of non-disjunction is happening. That means basically the chromosomes which are there, they should move away from each other when it divides into 2 cells, but over here it's not happening properly. Some will get extra. When you give someone extra, you're taking away from someone else as well, right? So someone gets extra, but over here the cell is getting less number of chromosomes as well. This condition is known as aneuploidy. Here an organism has an abnormal number of chromosomes, either due to gain or loss of one or more chromosome. It is caused by non-disjunction during what? Cell division. There are 2 examples: monosomy, subcategories, okay, monosomy and trisomy. Monosomy means basically what? You are losing one chromosome out there. Trisomy means you're gaining one extra chromosome out there. There are 3 chromosomes. For example, Down syndrome. You know, in Down syndrome what happened? There is 21st chromosome. 1. Yes, Poma, you're right. In Down syndrome, you get this chromosome from one parent, you get this chromosome from another parent. Okay, both of them are giving their copies of 21st chromosome. But now what happens? From one parent you're getting one more extra copy. So this is known as trisomy. Now over here these 2 chromosomes were enough for production of proteins. But over here one more guy has come, and do we need it? No. Obviously this will show some symptoms. That person will be having what? Down syndrome, which is known as trisomy of chromosome number 21. So in total how many chromosomes will be there? We generally have 46 chromosomes. One extra chromosome comes in, there are 47 chromosomes in Down syndrome patients. Okay, you need to know the symptoms of Down syndrome. Please go through it once. Okay, there will be palm crease. Okay, there will be stunted growth. Things like that you have to remember. Very, very important. Now, Turner syndrome is an example of monosomy, where I told you from one parent you should get X chromosome, from another parent also you should get X chromosome for being a female, but from other parent you're not getting anything. Only one X chromosome is there in one. For example, a sperm came with an X chromosome, but the egg is not having any chromosome, any sex chromosome. So in this condition, it is XO condition. So how many number of chromosomes are there in total? We just have 45. No 46, it's just 45, my dear children. Got it? Okay, now there is one more example, that is Klinefelter syndrome, and you need to know about them. And they have asked you about the karyotype of Klinefelter syndrome. Do you have any idea what does that mean? Karyotype means the chromosome pattern and everything. So I'll tell you, in Klinefelter syndrome, a person should have had one X and one Y. But a person is having an extra X chromosome. Extra X chromosome. So in general, my dear children, we have 44 autosomes. Autosomes are the ones that is not responsible for our sex. It is just there. Okay, 44 autosomes are the ones that is responsible for all the other characteristics of our body. Now we have X chromosome, that is XY or XX. Right, this is how it goes. So this is the 45th chromosome, this is the 46th chromosome. But over here it is XXY. So 44 autosomes plus XXY condition. So how many chromosomes are there? 44, 45, 46, 47. In total 47 chromosomes are there, and this is the karyotype. This is how you can write the karyotype. Or you can also write it one more way, that is 47 XXY. Use the saying that 47 chromosomes are there, and why it is 47? Because one extra X chromosome is there. You can either write like this, you can write like this also, but make sure you're writing autosomes over here if you're writing like this, and it should come first, and then all this thing should come. Okay, cool? Yes, Nagaraj, absolutely correct. Okay, so what are the symptoms over here? So it had to be a man, but actually over here, since there's an extra X chromosome, they will show the female character. For example, gynecomastia. You will get this question. Which is the disease that shows gynecomastia, that is development of breast in the case of males? It is Klinefelter syndrome. Underdeveloped secondary sexual characteristics. Okay, because it is not. As you can see, one extra chromosome is there that hampers everything. Do you think sperm formation takes place over here? No. Infertility will be there because of lack of spermatogenesis. This person will be tall with long arm and legs, and there will be learning difficulties. You don't need to learn that. At least these 4 symptoms would be enough for you guys. Done? So please make sure you're going through all of this. Now, what is incomplete dominance? Explain it with the reference of flower color in Snapdragon. Do you need me to explain this, or can we skip it? Incomplete dominance means what, my dear children? Where a gene is not completely dominant over the other one. That means, for example, over here, I have taken capital R, capital R. This is red. Capital R stands for red color. Okay, this is small r, small r. Small r stands for white color. So this is red color plant, white color plant. I am crossing them. So capital R, small r, small r. When I cross them, all of them, my dear children, would be like this. Now, what is the color that should be shown? What is the color that should be shown? So over here, capital R, if at all it was dominant over small r, it should have been showing red color, but that's not the case. It is showing pink color, which means what? It is incompletely dominant. It's not completely dominant, it is incompletely dominant, my dear children. This is known as incomplete dominance, which you see in Snapdragon or also in Antirrhinum. Okay, you also see it in Antirrhinum species. The same thing, Snapdragon or Antirrhinum, that is what you call. Now, what did they do the next, my dear children? They took the capital R, small r plants and they crossed it. Okay, when they crossed it, they found that capital R comes together, you get red flower again in the next generation, F2 generation. Capital R, small r, you get pink. Capital R, small r, again you get pink. Small r, you get white flowers. So if you see, my dear children, 1, 2, 3, they are all what? Are they pink? No. Are they red? No. Only one is red. Where both the capital R are there, it is red. But over here, 2 of them are going to be pink and 1 is going to be white. So what is the phenotypic ratio? I see 1 is red, 2 are pink and 1 is white. So phenotypic ratio, the physical appearance ratio is 1:2:1. Okay. On the other hand, if you're asked about genotypic ratio, so again, capital R is one genotype. Capital R, small r is second genotype, and small r is another genotype. So here you see 2 capital R, small r, which is why I'll again say 1:2:1 is the genotypic ratio. So over here, phenotypic and genotypic ratio both is the same. It's 1:2:1. And whenever you write phenotypic ratio, you have to mention that 1 is what? Red. Pink, 2 is what? Pink. And again, 1 is what? It is white. When you write genotypic ratio, you have to mention 1 is what? Capital R. 2 is what? Capital R, smaller. 1 is what? Small r, smaller. Clear? Exactly, Nagaraj and Poma, all of you doing great. Fine? Cool. Now going ahead with the next question out there, write the schematic representation of one gene inheritance in Pisum sativum. One gene inheritance in Pisum sativum. This thing is basically simple. When you're talking about tall plants and dwarf plants, you're talking about one gene. This gene is coding for height. This is monohybrid cross. That is what you are asked about. So you have to mention that. So in the first, what happens, my dear children? So over here, from this parent you get capital T. From this parent you get small t. Make sure you're writing parents and the gametes and everything out there. Now when you cross this and this, you get capital T, small t. All of them are going to be tall. Why? Because you have a capital T allele. It is going to be dominant. Here it's not incomplete dominance, it's fully dominant. Okay. So all of them are going to be tall. But the genotype is what? Capital T, small t. Now Mendel was not satisfied. He did a cross, self cross. And then when he did that, my dear children, what did he find out? Capital T, capital T come together, here it's tall. Capital T, small t comes together, again it's tall. This and this, again it's tall. This and this, my dear children, it will be dwarf. So, but if you just see the phenotypic ratio, 1, 2, 3 looks to be tall and 1 is dwarf. So what is the phenotypic ratio? It is 3:1. What is the genotypic ratio? 1 is capital T, capital T. 2 of them are capital T, small t, and 1 is small t, small t. So 1:2:1 is the genotypic ratio. So when it is incomplete dominance, genotypic ratio is 1:2:1 and phenotypic ratio is also 1:2:1. But over here, phenotypic ratio is 3:1 and genotypic ratio is only 1:2:1. I hope it's clear. So you have to mention about all of it. This is known as Punnett square. It was given by Reginald C. Punnett. He was a British scientist. Okay, he was a British scientist. Please remember that also. Apart from that, you can get one more type of question. I think I have officially completed. Is it? No. Okay. We have few more things. Chalo. Okay, let's go ahead with the next one. Write a short note on sex determination method in the case of birds. Shall we go ahead with that? What happens in the case of bird? What is this type of sex determination that you see over here? So my dear children, in the case of birds, you have ZZ and ZW. Which one is female, ZZ or ZW? It is this one which is a female. For the first time, it shows female heterogamety. Okay, female heterogamety means they are going to produce two different type of gametes. One is Z, another one is W. Here, the males are going to produce one type of gametes, that is just Z. Now over here, how do you show sex determination? This and this you have to cross and show it is Z and Z. What is it? It is a male. This and this, it is what? ZW, it will be female. This and this, ZZ, it's a male. This and this, again ZW, it's a female. So this is an example of female heterogamety. Explain like that, make it like this, and that's all. Only example in the case of birds. Okay, so birds are the only ones that shows female heterogamety according to your NCERT. Cool, works. Let's go ahead to the next question. So 2 mark done there. What is haplodiploid sex determination mechanism? Very simple. What do you mean by haplodiploid sex determination mechanism? Haploid means what? Half the number of chromosome. Diploid means what? Double the number of chromosome. Yes, Nagraj, you're correct. Okay, so over here, always remember, females are diploid. They are diploid, that means they have 2n. And males are haploid, that means they have half, that means n. So 2n means, suppose it is 32. Then what is n over here? It will be 16. Half of 32 is what? 16. Right. Now, basically what happens, here you have the female with 32 number of chromosomes. Right. She will undergo meiosis. Why? To produce gametes. Now in the gametes, since it is meiosis, in meiosis you have reductional division. The number of chromosomes becomes half, it gets reduced. So in one gamete she has 16, another gamete also it's 16. But the males, they were already 16 in number. The number of chromosomes are already 16. Will they undergo meiosis? No. They undergo mitosis. They just increase the number of cells, that's all. So they undergo mitosis, and again you have a gamete which has 16 number of chromosomes. Suppose this gamete of the female and this gamete of the male comes together, you have 32. What will it be? It will be female, because 32 is found only in the case of females. Suppose one particular gamete out there which was produced by the female, it did not undergo fertilization, and without undergo fertilization it will become what? Male. And you have a specific name for this process, is known as parthenogenesis. Without fertilization, when a male is being formed, this is known as partheno. It's not partheno, it's partheno. Parthenogenesis. It is found in the case of honeybees. Okay, so male drones are being produced, and over here females can be workers or it can be queen bee also. Fine, this is known as haplodiploid sex determination. So diploid ones are female and haploid ones are males. Now you tell me, will this particular child have a father? Does it have a father? Yes or no? In the chat section right now. Does it have a father? The male bees which are produced, does it have a father or not? Simple. Come on children. Hype up, hype up. No, no, no. Yes, exactly, no. One more question. Does it have a grandfather? Father is not there, agreed. Does it have grandfather? Hello, R.A. Tidagundi. Okay. Does it have a grandfather? First. Grandfather, yes or not? Yes, grandfather will be there. How? This female is produced with the help of male and a female both, right? So grandfather will be there, but father is not there. This is given in your NCERT, okay? So father, in the case of male drones, father won't be there, grandfathers would be there. So grandfathers would be having grandsons, but fathers would not be having sons. That is also being mentioned. So I just wanted to help it being cleared in your head. I hope it's clear. Now 25th question, we have the blood group questions, very interesting questions. Okay, so over here, write the genotype of the parents when the children are with AB and O blood groups. So all the different type of blood groups are there. Okay, it can be A, it has B, it has AB, one child is having AB, another child is having O also. How is it possible? How is it possible? In this particular condition, if you want all of them, my dear children, it should be IA, IO. One parent should be like this, and the other parent should be IB, IO. Why do I say so? Because for O blood group to come, both the parents should have at least one allele like that. Then only O will come, because O is what? Recessive. Then, so this and this comes together, that's when the child will be IO, IO. Okay, now for A, B to come, from one parent you should get A, from other parent you should get B also. So that's why I wrote here A and here B. So when this and this comes together, you get what? AB blood group. IA and IB. Now for A blood group to come, this one and this one can come. One A and O. A is dominant over O, my dear children, which is why you will have a child with A blood group as well. Similarly, one B and one O. So IA, IO. So IA and IB is also possible. Wait, I'll write it. IB and IO. Here, this is dominant over this, which is why B blood group is possible. Okay, so the genotype of the parent blood group can be IA small I. Small I means IO only. Okay, so that is being mentioned over here. Done? Deal? Ready? Next question. Define point mutation. Give an example for point mutation. Tell me. What do you mean by point mutation? When mutation is happening only at a single base pair. We studied in sickle cell anemia. It was GAG. It is replaced by A is replaced by U. This is point mutation. It is happening only in one single point, and this is changing the whole thing. So give this example and tell. A comes before U, so you need to remember like that. Similarly, glutamic acid is replaced by valine. So G comes before V. So it was glutamic acid and then it formed valine. These are amino acids, and amino acids, when they come together, they form proteins. And the protein which is going to be formed because of this mutated amino acid, my dear children, it will be different from the normal protein out there. Cool? Yes. Next. What are the conclusions drawn by T.H. Morgan from the crossing experiments in Drosophila with respect to linkage? What did T.H. Morgan say with respect to linkage? He said that if recombination is more, then what? The distance which is there, it will be less or more. If recombination is more, they are forming new combinations out there, so distance between them would be more or less? It would be more. They are loosely linked. If recombination is less, then both the genes would be tightly linked. This is what he said, and we already discussed it before, so I don't think we have to discuss it. Anyone who thinks that you need a repetition, you have joined the class right now, you can tell me now. Okay, otherwise we'll go ahead. Come on. Just let me see the okay. I'm not able to tightly. Okay, I think slowly it is coming. Is the network slow by any chance? Children, I hope you understood it. We discussed it before also, so I'm not putting too much time into it. If at all you think you need an explanation, you let me know. Okay, now last question of this particular chapter. What is test cross? Write its significance. What is test cross and what is its significance? Do you have any idea? Test cross basically means, suppose I have a plant which is tall. Now, ma'am, is it tall like this or is it tall like this? How do I know it? Whether it is heterozygous or homozygous. How will I get to know this? Only if I do the test cross experiment. What do you do in test cross? Basically, you are going to take a recessive parent. What are you going to take? You are going to take a recessive. Please understand, it is recessive for sure. And you're going to cross them. What? I didn't get you. I'll tell you. So small t over here, capital T over here. Similarly, another scenario, small t and capital T over here. Now, if I cross them, what do I get? This and this capital T small t, this and this again capital T small t, this and this capital T small t, this and this capital T small t. Ma'am, all of them are tall. Ma'am, all of them are tall. Okay, fine. I got it. Now let's go ahead. So over here, when I cross capital T small t, it is capital T small t again, When I cross this, it is small t and small t small t. So two of them I can clearly say they are tall because they have one dominant allele, and two of them are dwarf. What do I understand from this? That the plant which was appearing to be tall, it probably can be this or it can be this. So for understanding the genotype of a plant which is showing the dominant trait, which can be tallness or anything else, we do a cross with the homozygous recessive individual or recessive parent, and this is known as test cross. You are testing what is it, okay? And why do you think you need it? Because suppose I want to I am doing a crop breeding program where I want only tall plants, so basically I'll take this particular plant. No, I won't take that plant, because in the next generation I might find some dwarf plants. I do not want them. Got it? So this is known as test cross. So since you're going back, this is also known as back cross. Okay, since you're going back and crossing, this is known as back. This is a type of back cross. This is not exactly known as back cross. This is a type of back cross. Back cross can be when you go back and cross with either this or some other parent. When you're specifically crossing with this, you call this as test cross. Cool? Done? Clear? So now, before I go ahead with molecular basis of inheritance, okay, you have to give me a thumbs up if you understood everything. You have to stop me right now and ask your doubts if you have any doubts in principles of inheritance and variation. Can we get that? Can I know that? I deserve to know that, children. And if it's all cool, you understood everything, apart from giving thumbs up, click on the like button also on the video. Harshvardhan is saying thumbs up. Thank you. So that means Harshvardhan is all clear. Everyone else, children? Evolution is not being taught here, my dear child Poma. Since evolution was not there for everyone, I didn't take it. If you want, I can include it towards midterm when we do another live, okay? Nagaraj, okay, thank you for telling me that. Okay, children. So I am going ahead. You. Okay, is there for us. Poma, I'll try to include it in the next live, okay, whenever it's there. Ma'am, what are the chapters included in this? So, principles of inheritance and variation, molecular basis of inheritance, human health and diseases, human reproduction, sexual reproduction, and flowering plants. These are the chapters which are included. Okay, now going ahead with the next set of questions out there. Okay, fine. Molecular basis of inheritance, my dear children, you will get two long answers and one 2-marker and also one MCQ from this question. So we have to make sure we complete all of it. Okay, just a second. I am doing some mistakes out there. I'll log in again. Okay, try to do this question and we will be back. The number of nucleotide pairs present in the DNA of primary oocyte of a newborn in human is what? The number of nucleotides pairs present in the DNA of a primary oocyte of a newborn is what? Come on. A. Is it A? Let's see that. I think I accidentally touched my hand and some message has gotten deleted. I'm so sorry for that. Okay, so which is why even I'm getting, okay, fine. So the number of nucleotides present in the DNA of primary oocyte, those of you are saying 3.3 into 10 to the power 9. I'll say it is wrong. Why I'll say it is wrong? Why do you think it's wrong? Primary oocyte, my dear children, if you have studied human reproduction chapter, primary oocyte is still having 2N. This primary oocyte has just begun to have meiosis and it got stuck. Later, when it's complete meiosis, you get one first polar body and the secondary oocyte. So this becomes half. So over here it is diploid only. And the diploid number of chromosomes, my dear children, it is 6.6 into 10 to the power 9. Okay, it is 6.6 into 10 to the power 9. Fine. Sir, it will take probably 1 hour.
1 hour.
Okay, so it is 6.6 into 10 to the power 9, not 3.3 into 10 to the power 9. If at all you were asked about secondary use side, then the answer would have been different. Cool? Works? Okay. Okay, cool. I'm really sad. I think I have accidentally blocked someone. Okay, anyways, a DNA segment has a total of 1000 nucleotides, out of which 240 of them are adenine containing nucleotides. How many pyrimidine bases in this DNA segment does this DNA segment possess? So over here, you're given the total number. 1000 nucleotides are there. Okay, and out of that, 240 of them, you know it's adenine. And what is adenine? It's a type of purine. Apart from adenine, what different type of purine do you have? You have one more type. That is what? That is guanine. Guanine is also purine. Okay. And now, apart from all this, you have something that is known as pyrimidines. 2 of them. That is T and C. So A binds with T and G binds with C. That is a rule. So how many number of adenine is there? That many number of thiamine will also be there. But you were asked about what is the total number of pyrimidines. How do you find it out? So my dear children, 240 here, 240 here. What is the total? It will be 480. It will be what? 480. Now in total how many is there? 1000 is there. So 1000 minus 480 if you do. Okay, 1000 minus 480 if you do, what do you get? It comes out to be around 520. So 520 is there now. 520 is left, and now what do you have to do? You have to divide between G and C. So for that you can just divide by 2. And when you divide by 2, it comes to be 260. So this is 260. And if this is 260, this also is what? 260, my dear children. Now if you calculate the number of pyrimidines, 240 plus 260 comes out to be what? 500. So your answer would be B. Okay, your answer would be B. Fine? Cool? Guess. Got it? Children, are you getting it or not? I'm assuming you're getting it. So your answer is 500. Look for the question carefully and then only solve it, which is why I've put this question. Okay. Now, next thing. The 2 nucleotides of DNA are linked through 3 and 5 carbon of pentose sugar by what? So 2 nucleotides, this is 1 nucleotide, this is another nucleotide, and they are linked to 3 and 5th carbon. So 3rd carbon here and 5th carbon over here. This is the 5th carbon and this is the 3rd carbon. What do you call this bond as? So over here you see in between you have phosphate. Here carbon and phosphate is being bonded. Here also carbon and phosphate is bonded through oxygen, What do you call this bond as? This is known as phosphoester bond. What do you call this? Phosphoester bond. And when there are 2 phosphoester bond, you will call this as phosphodiester bond. So your answer would be what? B. Yes, exactly, Pama, exactly, Nagraj. Correct. Great going, my dear children. And also Harshvardhan, I couldn't see that, your previous answer, but thank you so much for replying. Next question. GGU, GGC, GGA, GGG all code for glycine. Which salient feature of the genetic code does it prove? So you need to know all the features of the genetic code. It is very, very, very important. So what feature does it show? All of these are coding for one amino acid, that is glycine. So over here, when do you call it this? When more than one codon can code for the same amino acid, you call this feature as what? Degenerate feature. So one amino acid can be coded by multiple codons. This is known as degenerate feature. So your answer would be what? B. Your answer would be B. Please remember, you need to remember all of this thing, that it is going to be universal for all organisms. It is going to be triplet. 3 codons are going to be there. Unambiguous means what? One codon codes for just one amino acid. That means GGU cannot code for anything apart from glycine. That is what you mean by unambiguous. They are non-overlapping. As you can see, they are different. They do not have any commas. In general, when you read mRNA, they do not have any comma. It goes straight like that. Okay, start codon is AUG. It codes for methionine also. There are 3 stop codons, UA, UGA and UAG. So UAA, UAG, UGA, 3 of them are there, they are stop codons. Done? Good going, Nagaraj, Poma and Harshvardhan. All of you doing great. Where are the other children? So I see 12 people are watching this video but not replying. Why so? All my superheroes and heroines, please answer. Okay, so amino acylation of tRNA occurs during. What do you mean by amino acylation? The tRNA which is there, no, it is responsible for reading the mRNA's information. Okay, it is going to transfer some information out there. So the tRNA which is there, it has to be bonded to one amino acid. This process is known as amino acylation of tRNA. So on the mRNA, some codon will be there, for example, GAG. So what will be the amino acid over here? It will be glutamic acid. So who combines it? This process is known as amino acylation of tRNA. Okay, Trishank is there. Good to be back. Yes. Yes, Nagraj. And yeah, the answer is B. Okay, amino acylation of tRNA occurs during translation. This process is translation, where you are translating the information present in the mRNA to form the proteins out there. Cool, Poma, you are also correct. It's not regulation of operon, nothing like that. So let's go ahead with the next question. Otherwise, we might get late. If a processed mRNA of a particular cistron contains 636 base pairs. Now this you have to attempt and you have to let me know, okay? Be very careful with this question. This is a tricky question. If a processed mRNA of a particular cistron contains 636 base pairs that codes for a polypeptide chain, what are the number of amino acid that it codes for? So there is a cistron. That means a unit of structural gene, or the gene out there. Okay, and there are 636 base pairs. All the base pairs are like this, and they are going to code for what? A polypeptide chain. How many amino acids do you think they are going to be there in this particular chain? 6B. Oh my God, did you crack it actually? So my dear children, 636 means these base pairs are in the form of codons, triplet codons. So if I divide by 3, it will be what? 212. Right, now you might see answer 212, this is the answer. But no, it is not. Why? Because one particular codon which is there, one particular codon, that means 3 base pairs at the end, they are what? Stop codons. They do not code for any amino acid. So total number of amino acid you have to minus 1. Okay, so it will be 211. Your answer would be B. Kudos to you guys. Nagaraj, Poma as well as Trishank. Superb. Okay. So your answer is 211. Make sure you are deleting that particular thing. I have written over here. So if at all you get the PDF, you will be able to know that as well. Next question. In pea plants, each character is governed by a gene which is monocystronic, polycystronic, operon, intron. What do you think the answer of this question is? In a pea plant, each character is governed by a gene which is monocystron, polycystron, operon, intron. So pea plant, my dear children, it's a eukaryote or a prokaryote. Plants are eukaryotes. They have nucleus, they have nuclear bound membrane, they have organelles, they have everything out there, just like a eukaryote. In the case of eukaryotes, you know, the structural gene the cistron will be having only one gene. This gene will be coding for one particular protein. In the case of prokaryotes, for example, bacteria and all, know what happens? Suppose this is a segment. Over here they will have structural gene 1, then there will be another gene, then there will be another gene. This codes for one protein, this codes for second protein, this codes for third protein. This is known as polycystronic. Okay, they have multiple cistrons, but over here there is only one cistron having one particular gene. So your answer would be monocystron. Okay, okay. Chalo, next. Great. Good to see you guys answering the questions. Now, match the following. I put this question because I just wanted to know general thing. Okay, so that you remember something very important out there. So the way I solved this question is that I know lac operon which was there, it was given by Jacob and Monod. Very much easy for me to remember. And I also know Griffith was the one who did the transforming principle experiment. With just this information, can I find the answer? Yes. So C goes with 1. C goes with 1 over here and here. So I can eliminate these options. Griffith, transforming experiment. D goes with 2. D goes with 2. I just have one option and that is this. And hence I got my answer. Okay. So over here, Alec Jeffrey, actually he was the one who gave the DNA fingerprinting. You can remember Jeffrey F. Finger, that sound is there, no? So fingerprinting, okay. Now F. Sanger also sounds like the same, but please remember F. Sanger, S is for sequencing. Okay, DNA sequencing. He was the one who gave sequencing. Done. I know sometimes I do not make any sense, but I definitely know, I know one more thing as well, that is you will remember it anyhow. Okay, so these are the things out there, and are we good to go with the next question? Question number 9 out there. Human insulin is produced by E. coli cells by rDNA technology. This proves that genetic code is what? So over here I am saying the human insulin, which is produced in human beings, it is now being produced by E. coli, my dear children. That means they also are producing, they also have the same translation method in their gene also. The same codons are there, the same codons are coding for the same amino acid. What does this show? In E. coli and in us, it's same. That's a bacteria. We are different. Yes, 9A. Yes, Sperma. Yes, Nagraj. It is universal. The genetic code is what? Universal. It will be same for all the organism, except some codes. Okay, mitochondrial codons will be different. Protozoan codons are different. There are 2 exceptions. Rest everything we can consider it to be the same. Okay, done? Fine. Next question. Now these are 5 marker questions. Again, we are entering into 5 markers and then 3 markers. So please make sure you are listening to me carefully, and then we'll go ahead with the next chapter. So from now on, I'll make up the video a bit more faster if it's okay for you guys. It's okay? Can I? Give me a thumbs up if I can. If you want me to go in this space itself, let me know that as well. Come on. I'll just wait for you to answer and then we'll go ahead, okay? So the first question says, list out the salient features of the double helix model of the DNA. So you have to talk about the DNA as a whole. Okay, so over here, my dear children, you have a pentose sugar, first thing. So you try to answer, by then I'll make the thing. So this is a pentose sugar, that means it has 5 carbon. First carbon is over here, second is over here, third is over here, fourth is over here, fifth is outside. Okay, the first carbon is attached to a nitrogenous base. The 5th carbon is attached to phosphate group. This bond is known as phosphoester bond. This bond is known as N-glycosidic linkage. Okay, N-glycosidic bond out there. Now basically what happens is that, suppose there is one more nucleotide. This whole thing is known as a nucleotide. A nucleotide is made up of pentose sugar, phosphate group and a nitrogenous base. Okay, now this nucleotide is connected to some other nucleotide. Here the 3rd carbon, my dear children, is attached to this phosphate group. This is what you call as phosphodiester bond. Here phosphoester bond, here also phosphoester bond, 2 bonds coming together, phosphodiester bond. Right? Now my dear children, what exactly happens is that the 5th carbon, suppose it's free over here. So I will call this as 5' end. And over here it is still attached to what? Nitrogenous base. On the other hand, if you see, the 3rd carbon is not attached to anything. So this is the 3' end. So over here, if I make the DNA, it's 5' end and it is 3' end. Now the other strand, the double strand, it has to be double stranded, right? The other strand runs in opposite direction. It will be 3' and here it will be 5'. So I'll say that it is anti-parallel. It's not just parallel, it is anti-parallel. They are ulta. Okay, and in between that you have nitrogenous base. The nitrogenous base that you see, they are, so this trans nitrogenous base and this trans nitrogenous base, they are going to come together with the help of hydrogen bonds. Okay, so over here A bonds with T with double bonds and 2, you can remember T for 2. Now G with C with triple bonds. G and C look so much alike, so they are very close to each other. Okay, so these are pyrimidines and these are purines. In the case of RNA, instead of having T, you have uracil. Instead of having T, you would be having what? Uracil. That is about RNA. We'll talk about that later. So these are the features. Now, how do you write it in the form of the answer? That also is important. So DNA is double helix with 2 anti-parallel polynucleotide chains. Polynucleotide chains means nucleotide coming together making polynucleotide. Chains are coiled in right-handed manner. They are coiled like this on the right-handed side. Phosphodiester bonds link nucleotides in 5' to 3' direction. Okay, in 5' to 3' direction in one strand and 3' to 5' direction in the other strand. So anti-parallel. Hydrogen bonds between nitrogen bases hold the strands together. Adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Now, over here, Chargaff's rule is there. Purines A and G is equals to pyrimidines T and C. That means if at all 10 number of adenine is there, that means what? Thymine will also be 10. Why? Because they are pairs, right? Similarly, guanine, suppose it is 10, here cytosine will also be what? 10. That is Chargaff's rule. Now, one more very important thing. That is this. Listen to me carefully. Okay, so over here you have the DNA. This whole thing is known as one helix. This is one helix. One helix has how many base pairs? 10 base pairs. 10 base pairs are there in one helix. What is the distance of this whole thing? It is 34 Armstrong. It is what? 34 Armstrong. So in between this, what is the distance, ma'am? If the whole thing is 34 Armstrong, in between this 34 divided by 10, it will be what? 3.4 Armstrong. Ma'am, we do not have to study in Armstrong, we have to study in nanometer. Then 34 Armstrong means 0.34 nanometer. So what is the distance between this then? It will be 0.03. Wait. Wait. Sorry. I'm so sorry. I did a mistake. Forgive me. So 34 Armstrong means 3.4 nanometer, and over here the distance between this would be 0.34 nanometer. Fine. This is the only thing that you need to remember. Now one more thing, pentose sugar and phosphate form the backbone of the DNA. So the pentose sugar which is there and the phosphate which is there, it forms the backbone, because if the phosphate is not there, another pentose sugar will not be able to join. If pentose sugar is not there, there won't be any linkage between the nucleotides out there. So both of them are going to form the backbone of the DNA. These are the points that you should definitely be writing down. If at all you need, you can just make 2 of them like this. That's enough. Cool. Now going ahead with the next question out there. What is it? Describe the steps involved in DNA fingerprinting technique. Now this is very interesting and this question can be repeated. Okay, they can ask you, okay, I'll tell you. So DNA fingerprinting is where, for example, there is a crime that happened. I did it. I am the criminal. But my friend was also there. He is also a suspect. Now basically what is happening is that my blood stains are there in the crime site. That was taken from the blood, my dear children, the DNA was being extracted. Basically the cells which are there, from that the DNA is extracted, right? Now the DNA which is there, I know everything about this DNA. I tried to code it up. Not me, the person who is investigating, they tried to code it up. So there is a sequence and everything out there. Okay, now they have a doubt on me and my friend. So both of us were called. Our DNA was also taken. When our DNA was taken, my dear children, what exactly happened is that both of our DNA was cut with the help of same enzyme, which is known as restriction enzyme, my dear children. Okay, so I cut this DNA with the help of restriction enzyme. Now I have small strands over here. Small DNA strands. Similarly over here as well. After that, these DNA strands were made to run in a gel electrophoresis chamber. So over here, this is person A, this is person B. This is mine and this is my friend's. So this was the thing that I observed. The DNA, which was smaller, they run faster. The ones that are larger, they run slower. Okay, and over here the winding pattern was something like this for person B. Now the DNA that I got from the criminal side, the crime side, my dear children, when electrophoresis was performed, when same restriction enzyme was used to cut it, and when electrophoresis was performed, I found the same type of banding, just like man. And then I got to know that not me, but the investigators got to know that I am the criminal. But it is not that simple. How do you observe this band? First of all, you make it run to the gel electrophoresis chamber, and then you use a nitrocellulose sheet. This sheet has been put like this. So everything which is there on the gel electrophoresis chamber, it comes over here. So the DNA strands are now over here in this particular sheet. Still, will I be able to view it? No. I need to basically make certain probes run through it. Some tagged probes out there, which means they would be showing up color when I put it under the UV radiation. So these are tagged probes out there. Where do I get these probes from? From here itself. So over here, when the criminal side, the DNA that I got, okay, for suppose it was G, A, G, C and things like that in my DNA. I made a complementary strand out of it. It was C, T, C and G. So this is the probe that I have. And I made it tag with a fluorescent stuff out there. So this is a fluorescent probe that I have. Now when I made it run through the nitrocellulose sheet, it will only go and bind to the places where it has the complementary pair, and which is in mine, my dear children. It will go and bind in my dear region. Here also there might be some things that are common, it can go bind there also. But anyway, when I see that this, then later when I put it under UV radiation, I find that both of them are actually same. And with that I get to understand that I am the criminal, not me, but the people out there. This is known as DNA fingerprinting. All of our DNA are very, very unique. Now the same thing can be done with the help of VNT, or variable number of tandem repeats. So in my body, there are certain unique things which are repeated 10,000 times. For example, GAGC. It is repeated 10 times. But in yours, it might be repeated multiple number of times. So depending on that also, I can try to find out the same thing. So the same theory you are going to use. First, you're going to cut it with restriction enzyme, then electrophoresis chamber, then you are going to do blotting technique. Then basically you are going to run probe through it and then put UV radiation. You will be able to visualize everything out there. So same thing you have to write in the form of your answer like this. Isolation of DNA, digestion by restriction enzymes, separation by gel electrophoresis chamber, transfer to the membrane, hybridization with the DNA probes, and then visualization. So these are the steps of DNA fingerprinting, and you can write the same steps in VNTR question also. But there you are going to use variable number of tandem repeats, which is going to be very unique in me and you. Okay, the number of repeats. It can be 10, in mine it can be 20. Now answer the following question. Construct a complete transcription unit with a promoter and a terminator on the basis of hypothetical template strand given below. So this is a template strand. Okay, this is what strand? Template strand. And what do you mean by template strand? This is a strand which is going to undergo the process of transcription, my dear children. So from this DNA, I am going to get my RNA. What RNA? mRNA. So in which direction is it? It is in 5' to 3' direction. Always remember. Okay, it is in 5' to 3' direction. Now write the RNA strand transcribed from the above transcription unit along with polarity. So first of all, we have to make the full transcription unit. How can you do that? So over here, see at this particular picture. So I have put the whole strand over here, and then in front of that I made a box, wrote promoter there. After that I made a box and wrote terminator there. Similarly, I have to make a complementary strand in 5' to 3' direction. Okay, and over here with A, here T will be going T, here A will be going, like that I have to make a complementary strand, and I have to mention what? It is what? A coding strand. So instead of writing just promoter over here, I can just make a big box and write promoter, and here also I can write terminator, but you need to write promoter and terminator. That's when it becomes a whole, transcription unit. Also remember the template strand would be like this, 3' here, 5' there. Okay, 3' here, 5' there. Why? Because the new strand which is going to be produced, it is going to be made like this, from 5' to 3' direction. Why? Because RNA polymerase makes it from 5' to 3' direction only. And now, what is the strand that you get over here? It is this. How can you make it simply? So if at all you have made the coding strand here, you just have to replace the T with U, because this was DNA, the complementary strand, but you are making an RNA. So T will be replaced with U. So wherever you saw T, make it U, and here you have got the whole strand. Done. So this is the transcription unit, and this is the new strand that you have got. Clear? Done. Children, understanding? Not understanding? If understanding, give me a thumbs up. If you're not understanding, can you ask me? Come on, come on, come on, come on. Any doubts? Ask. No doubts. So I believe every one of you superstars, no doubts at all. And if every one of you are understanding so well, right? So that means I have to go to the next question. List the salient features of human genome. Now this is where I do not have to explain much. It's more kind of by hearting, my dear children. So human genome has these many numbers of 3164.7 billion base pairs. The average gene size, one gene size is usually made up of 3000 bases. But my dear children, there is one gene that is known as dystrophin gene, which is having 2.4 million bases. Okay, the total number of genes is estimated to be what? 30,000 genes. Out of this, 50%, so functions of 50% are known. Okay, functions of 50% are known and rest of them I do not know the functions. Less than 2% of the genome actually codes for the protein. In total, only less than 2% actually codes for protein. Rest of them, my dear children, are not coding for any protein out there. There are so many repeated sequences out there, and these repeated sequences are really important. Even though they do not have any coding function, they are important for understanding evolution and things like that. Okay, now this is important. Chromosome number 1 has the most number of genes, 2968. The first chromosome has the most number of genes. What is it? It is 2968. And the Y has the fewest, that is 231. Chromosome number Y found in the case of males, they have the fewest, 231 only. Now scientists have identified about 1.4 million locations where single base DNA difference is there. That means between you and me, all the DNA strand is same, just at one particular position it can be different. This is known as single nucleotide polymorphism. So these are the things. Everything which is mentioned over here is in millions, no trillions and no billions. You can remember like that also. Okay? Followed? Now going ahead, because that is your job to study it up, because there is nothing I can do to make you remember all of it. Okay? Next thing is lac operon, one very important concept out there. Listen to me carefully. So over here, there are two sets of genes. One is this is known as regulatory gene. Regulatory gene codes for a protein which is known as repressor. So regulatory gene can be negatively regulating, positively regulating. And Poma, thank you for telling me that. Okay, so here it is negatively regulating the production of one protein that is known as repressor protein. And this is the promoter sequence of this particular transcription unit. Similarly, you have another set of genes out there. For that, you have a promoter sequence, that is this. And this region is known as operator. So do you know when transcription begins? When a RNA polymerase comes, binds on this, and then it goes ahead. Okay. But over here, what exactly happens is that since this repressor protein is being made, this repressor protein goes and binds on the operator region. And when someone is coming and binding over here, do you think the RNA polymerase which will come and bind over here can go ahead with the transcription of Z, Y and A gene out there? Obviously no. And I'll say that this condition is what? Switch off condition. Over here, the lac operon is not working. These enzymes are not being produced. These particular mRNA is not getting transcribed itself. Now, on the other hand, this is in E. coli. In E. coli, this whole thing is happening. Okay. Now, E. coli is grown in a medium that has lactose in it. Just suppose it is grown in a medium that has lactose. What exactly happens is that this lactose enters the E. coli, and this lactose goes and bind on the repressor. Now the repressor is not free to come and bind over here. Okay, it is not free to come and bind over here. Now do you think RNA polymerase will come and do the transcription? Yes. It will make what? 3 enzymes. Z codes for beta-galactosidase, Y codes for permease, and A codes for transacetylase. Everything will be possible. So what do I call this lactose which comes and binds over here? I'll call this as inducer. It is going to induce the process of transcription. Okay, so it can be lactose or allolactose. Now what does beta-galactosidase do? It actually, so lactose was a good guy, but over here beta-galactosidase breaks down this lactose to glucose and galactosidase. So that is the thing. Why? Because the E. coli needs energy. That's why. So lactose is the energy medium. So lactose is broken down by beta-galactosidase to what? Glucose and galactose, and that's how E. coli would get energy in a lactose medium. On the other hand, permease is the one that will increase the permeability of the walls of the E. coli so that things can come in and go out. Okay, transacetylase is there to deal with all the other things which comes in between. Fine. Got it? So this is known as switch on condition of lac operon. So you have to make it like this and you have to explain. Always remember it can be lactose or even allolactose which acts as an inducer. And please write your answer like this. Switch off mechanism. When does it get switched off? What exactly happens when lactose molecules are absent in the medium? That's when we do not need the enzymes to be produced if lactose is not there, right? We need it when the lactose is there. So that's when it gets switched on, and you have to talk about each of these enzymes. Very, very important. And Poma, thank you so much. Your teaching helps us a lot, ma'am. It's so sweet of you to tell that. Thank you so much. Lots and lots of love. to all of you. Okay, question number 50. DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl. So always remember, these experiments are important. First experiment, DNA has semi-conservative mode of replication. Second experiment, DNA is the genetic material. So all of it very, very important. So who did the experiment? Michelson and Stahl. What experiment did they do? So they said that, suppose over here, this is the parental DNA strand. Now this parental DNA strand, when a new copy of DNA has to be made, what exactly happens is that both of them, they unwind. Now we have two strands, right? We make a complementary new strand out there. But don't you think one strand remains the same? That is the parental strand. So I'll say this is semi-conservative replication. The one strand is still the same, the parental, and there is one extra new strand being formed. But how do you prove it exactly? So basically what he did do with that, he grew E. coli, again E. coli, okay, Escherichia coli, in a medium which has N15 isotope. What isotope? Nitrogen 15, N15 isotope. And nitrogen, you know, is a part of DNA. Nitrogen is basis, right? So DNA takes up this nitrogen and it is, labeled as N15. Now, basically this DNA which is there, if at all I go it around through the centrifugation cap, my dear children, what exactly happens is that it will centrifuge at the bottom level because it is heavy. This isotope is heavy. So the strands would be showing up like this. It is a heavy chain that we get. The gradient which is being used is cesium chloride. Okay, we use cesium chloride for centrifugation. Now, what did I do? I changed the medium. So instead of using N15 NH4Cl medium, I am going to grow it in N14 medium now. Now the new DNA strand which will be there, it will be taking up this N14, right? New DNA strand will be taking up the N14. So over here I get a new strand, one with N15 because it was the parental one, and the new one with N14. Similarly here also I get something like that. Now when I do centrifugation, I find that a hybrid thing is being formed here in the middle. In the middle, a new strand is being formed. So I get to understand something has changed. Now again, my dear children, again I let the E. coli divide. So in every 20 minutes they are going to do their division. Replication is going to get completed. Now again I will find the same thing. So one strand will be parental. This time one is N14 and the other one, the new one, is also N14 because we are continuing in an N14 medium. But here you have the older ones. One is N15 and one is N14. Get it? So still you will be finding the hybrid one. And now you will be finding the new one as well. And this proves the semi-conservative mode of replication. I hope it's clear. If it was not semi-conservative mode of replication, my dear children, if it was totally new, then you would not be finding a hybrid thing. You would be finding just this new thing that is popping up here. You get it? Since it is hybrid, you are finding this. Since it is semi-conservative, you are finding this. I hope it's clear. Done? This is known as Michelson-Stahl experiment, and you have to write your answer properly. So I have written a whole thing, but you can choose what you need to write from the PDF that you're gonna get. Works? Cool. Now the next question. Oswald Avery and others have continued Griffith's transforming principle to prove DNA as genetic material. Substantiate. Children, are you understanding? Can I have some likes out there? Can I have some hearts out there? That's when I get to know the flying hearts, whatever you do. No? Not understanding? Understanding? Not understanding? At least tell me you're understanding or not. So I saw only Poma is understanding. Rest 9 of you are just sitting over here and seeing the PPT probably, right? Is what I understand. Okay, I see hearts. Thank you so much. And children, I would not come and eat you. Please talk to me. I'm a very cool teacher is what I believe so. Okay. Aksa is saying, no, you're not understanding. Okay, I'll make you understand. You can ask me the doubt whenever you have it. Okay. Oswald Avery and others have continued Griffith's transforming principle experiment. So what exactly happened over here in Griffith's experiment? For those of you who are not understanding, I'll explain you. So my dear children, Griffith took 2 strains of bacteria. One was rough strain and the other one was smooth strain. And this name of the bacteria is Streptococcus pneumoniae because it causes pneumonia. Okay. So the S strain had an extra covering, extra polysaccharide covering, which made it what do you call dangerous, because if someone get infected with the S strain, that organism would be dying. So Griffith did the experiment, my dear children, where exactly what was happening is that he put the R strain first into the mice and he found that the mice lived. Why? Because R strain is not dangerous, it is rough one. But when he put the S strain to the mice, he found that the mice basically died because probably it was causing some trouble out there. Then what did he do? He heat killed this S strain. He treated it with heat, this particular bacteria. So I'll write heat killed S strain. And then he put it into the mice and he found that the mice lived because something was getting damaged over here because of the heat. Now one more thing was done. Okay, when this was seen, one more thing was done. Heat killed S strain plus some R strain which didn't cause any trouble before. Both of them were mixed and now that was put into the mice, and now that this particular mice died. It was kind of strange because as of now it was not dying. Here also it was not dying. But when both of them were getting mixed, it was dying. Probably because this particular strain was getting transformed into something else. What something else? S strain. But how can that transformation happen? There was something that was getting transferred, but what was it? He didn't know that. That could be DNA, RNA, protein, all of it. So Oswald, Avery, MacLeod and McCarty came into the picture to find what was exactly it. So what did they do is that they took the heat killed S cells, okay, the heat killed S cells, the one that didn't cause any trouble. They treated it with proteinase. Proteinase are enzymes that are going to damage the protein. Okay, they are going to damage the protein of the heat killed S cells. So the proteins are damaged over here. Now they are going to add up the R cells also. And then they are going to infect the, then they are going to infect the mice. But they found that the mice is still dying. The proteins were dead, but that didn't have any change. The proteins have no role in the whole story. Then RNase was being mixed. RNase means RNA, it is going to digest the RNA out there. Same thing happened. There was no transformation that was being seen. Sorry, there was transformation that was being seen. That means it was still forming S cells. That means nothing else which was getting transferred, that was not getting killed in any ways. Now the last thing that they did is they treated with DNase. The DNA is now going to get damaged over here. So when they did treat it with DNase, whatever DNA was there in the heat killed S cells, all of it got damaged. And now when I mix it with R cells, do you think it can convert the R cells to something else? No. So no S cells appeared when it was treated with DNase. That means probably DNA is something that was responsible for the transformation. Get it? This was said by Avery, MacLeod as well as McCarty. So they were in the verge to say that DNA is the genetic material, but people didn't believe them. So which is why another people, that is Hershey and Chase, had to continue their experiment, and they did this experiment to prove that DNA is the genetic material. So Avery, MacLeod, McCarty are the people in middle. I hope you got it. Aksa, is it clear to you, dear? Okay, so please write it like that. Here is the answer. You can write it, read it properly whenever you have the PDF with you, and I think that would work. Now the last experiment, as I said, was done by Hershey and Chase. Again, this can be a question. Okay, so over here, listen to me carefully. What exactly happened? Thank you, Aksa, for telling me that. Okay, so over here, the last experiment which was done, it was done with the help of bacteriophage. Always remember, they will ask you, what is bacteriophage? Bacteriophage is a virus. It's not a bacteria. It's a virus that affects the bacteria. Again, you are taking E. coli over here. You have the bacteriophage. One bacteriophage was labeled with S35, sulfur 35. Now sulfur is a part of protein. Okay, amino acids which are there, they can take up sulfur, and it is a part of the formational thing out there. So sulfur will be there wherever protein is there. So the protein, this particular capsid which you see, the covering of the virus that you see, it is made up of protein, which is why radioactive S35 thing was there in the capsid only. It was not there in the genetic material. Now we do not know what is the genetic material, but anyways. Now radioactive P32 was also taken, phosphorus 32, and we all know phosphorus is a part of what? DNA. But over here, it went and radio labeled this particular thing because it was being used, my dear children, in the genetic material. You had the P32 thing. Now both of them went and infected what? The bacteria out there, which is E. coli. So here also infection took place, and my dear children, here also infection took place. So here the genetic material was taken inside. So what exactly happens is that bacteriophage gives in the genetic material to the bacteria. Okay, and when and then it uses the machinery of the bacteria to produce more bacteriophages, more baby bacteriophages. It works like that. So over here the radio-labeled thing got in, but here the radio-labeled thing was outside. Okay, and now the second thing which was done was blending. Blending means basically you are separating everything, whatever is there on the surface, and then centrifugation was done. That means it was made to rotate like this, and then what was found? No radioactive detected in the cell. So it was found that no radioactivity was there in the cell. This particular thing, bacteriophage, was labeled by S35. Right, and no radioactivity was found there, but in the supernatant, in the watery liquid, my dear children, this radioactivity was found. And we all know that this particular thing is what you find in the pellet, in the supernatant, and this particular thing probably would be having protein, which is why this is getting radioactive. On the other hand, when you centrifuge this, you found that the cells were radioactive. That means what did you label with P32? It was the genetic material. So probably the genetic material was what? The DNA, which is why the cells were radioactive, but over here this thing was not radioactive. That is the supernatant, the liquid which was there, it was not radioactive out there. Got it? So this is the scenario that you have for Hershey and Chase experiment. The people who did the experiment knew that in the DNA there is phosphate, which is why they took phosphate over here. The people who did this experiment, they also knew that in the protein there is sulfur, which is why they tagged something with sulfur. So that they get to understand whether it's protein or whether it's the DNA which is getting transferred. So at the end they got to understand it's the DNA which is getting transferred. That's all. This is Hershey and Chase experiment. Done. Now the next question is question number 18. Explain the process of translation in protein synthesis. So what exactly happens in the process of translation, my dear children? Here there is the mRNA. This thing that you see, this particular structure that you see, it is the tRNA. The tRNA, all of them would be coded with what particular amino acid, which is right up over here. Right, and these are the things, that is the mRNA, right? So the mRNA is read by unique tRNAs, which are in turn associated with one particular amino acid. And here you have the ribosome. This is the smaller subunit, this is the larger subunit. The first thing that happens is that all the tRNA which is there, it is going to get amino acylated. That means one amino acid is going to attach with this particular tRNA. So the tRNAs are completely ready. Now comes the mRNA. Okay, mRNA comes into the picture, and the smaller subunit will come and attach to the mRNA, right? Suppose over here you have AUG, codon. Now the first tRNA will come. The first tRNA will come with amino acid, which is what? Methionine. Always remember, it will be always methionine, the first tRNA. Okay, here if it's A, then here it's U because it's an RNA. Here if it's U, then it is an A. Here if it's G, then it is C. So this is the anticodon and this is the codon of the mRNA. Now comes the larger subunit, and when you have everything, this forms the initiator complex. What does it form? Initiator complex out there. Slowly, my dear children, what will happen? The ribosome will move like this, and when they move like this, they are reading each and every part of the mRNA. Again a tRNA would be coming with another amino acid, and when it comes, my dear children, this amino acid and this amino acid makes a bond between them. This is known as peptide bond, and here they will get dissociated. It will get dissociated from the tRNA, and this amino acid will be linking together. The same thing goes on again and again, and that's how you get a huge strand of what? Amino acids. This is what you call as the polypeptide chain. And at the end, when you reach the stop codon, the whole process of translation would be stopping out there. So you have to write your answer like this. So first thing that happens is what? Activation or charging of amino acid. So amino acylation of tRNA happens where it's the process of attaching an amino acid to the corresponding tRNA. The tRNA has 3' end and 5' end. It attaches to the 3' end. Always remember. And here you need a lot of energy, that is ATP. Now what happens, the next thing is that initiation. mRNA binds to the small ribosomal unit. I already told the mRNA comes and binds to the small ribosomal unit. The start codon is recognized by the initiator tRNA, the first tRNA carrying methionine. The last subunit also assembles, forming what? Initiation complex. Everything is ready. Now elongation will happen. tRNA brings amino acid to the ribosome, matching codons on the mRNA through its anticodons. So their matching codons and anticodons will be coming up together. And in between that, there will be peptide bond formation happening. Always remember, the ribosomes will be moving from 5' to 3' direction of the mRNA. Then last, when a stop codon UAA, UAG, UGE is reached, translation stop, as the release factor will come and join. So at that particular point, what will come and join? The release factor, there's something like that, which will come and join on the stop codon. Always remember, no tRNA comes and joins on the stop codon because there is no amino acid which is coded by the stop codons out there. So only release factor will come and join, and that's all. And the whole process of translation would be completed by then. So please remember this particular diagram and remember the steps. Cool. Now, the 2 marker questions out there, we'll take it a bit fast. Which sequence of bases transcribed from DNA are found both in hnRNA as well as mRNA? What do you find both here and here? What is hnRNA? Heterogeneous nuclear RNA. So whenever I say that from a DNA, transcription happens and you get an RNA, it's not directly mRNA. You get hnRNA. How is this and this different? Basically, the hnRNA, my dear children, it will be having regions that codes for protein. You call these regions as exons, and there will be regions in between. Okay, you call these regions as introns. They do not code for anything. They're just there. So they are known as the whole thing is known as hnRNA. Now basically, whenever you want to make a protein, you do not need these guys out there. And who is going to help in the formation of proteins? mRNA. So what do? You undergo a process which is known as splicing, where you remove all the introns. So introns are removed, and now there is a proper thing. Apart from that, this particular hnRNA undergoes a process which is known as capping. Okay, so over here some groups are added. Similarly, there is a process known as tailing, where over here lots of adenine nucleotides are being added up. So capping is being done, tailing is being done, splicing is being done. At the end, you get this processed RNA which is known as mature mRNA, which is finally going to move out of the nucleus and goes to the cytoplasm, where it meets the tRNA, and the whole process of translation would be happening up. So the sequence of bases transcribed from dRNA that are found both in hnRNA and mRNA are exons. That is its common thing. And exons are the coding regions that remain in the mRNA after splicing, while introns, my dear children, are removed during the process of processing. Done. Okay, so you are asked what are the sequence found both in hnRNA and mRNA? It is introns. Sorry, it is exons. Okay, exons would be there, introns would be removed. Done. Going ahead with the next question, mention the levels where gene expression can be regulated in eukaryotes. The first level, I can let transcription to happen or not let it happen. So the first level is that. Second is processing level. That means I can let splicing, tailing and everything to happen, or I will not let it happen. Okay. Third thing, I can let the mRNA go out of the nucleus, or I'll stop it from going out of the nucleus. The fourth thing, I can let translation to happen, and I can stop translation to happen, either way. So these are the 4 levels in which regulation can be done in the case of eukaryotes. Can the same thing be done in the case of prokaryotes, my dear children? No. Why? Because prokaryotes, do you think they have separate nucleus and things like that? Everything basically happens in the cytoplasm itself. So this type of regulation is not possible in the case of prokaryotes. In the prokaryotes, they have operons and everything. Lac operon we studied, right? That is there in that particular case. Works? Cool? Fine. Next, write a note on the role of sigma factor and rho factor in transcription in prokaryotes. So first thing, in transcription, I told you RNA polymerase is involved. So RNA polymerase cannot do its work alone. It needs someone else, and that is what? The sigma factor. For initiating the process of transcription, you need sigma factor. Okay, so sigma factor comes and attaches on the RNA polymerase, and that's when initiation happens. And then later on the sigma factor goes off. And at the end, when the whole process of transcription has to be terminated, you need one more factor, which is rho factor. So you can remember, like RNA polymerase is crying, I want to go out of this, I want to go out of this. So rho in Hindi means cry, right? So the same thing is happening over here. So rho factor comes and binds with the RNA polymerase, and the whole process of transcription would be stopped right there. Cool. So sigma factor binds to the RNA polymerase, guiding it to the promoter region for transcription initiation, and later it detaches. And on the other hand, rho factor facilitates termination by binding to RNA and unwinding the RNA DNA. So at the end, you have to unwind the RNA and the DNA thing that has been formed, right? You just need the RNA strand. And that is rho dependent termination. That's all. Now VNTRs, I already told about them. VNTRs are what? Variable number of tandem repeats, and they are different in my case, in your case. For example, I might have GACC 4 times, but in your body it can be there, in your DNA it can be there 20 times. And depending on that, I can get different lengths of DNA. For example, in my DNA strand will be smaller because it's just 4 of them, but yours would be larger. And depending on the size difference, my dear children, I can separate it by using the gel electrophoresis, as we discussed in DNA fingerprinting. Same thing. Okay, but it's just depending on the length that we are able to segregate things. That's all. It's not too difficult. Okay, so that is VNTR. That was asked in July 2018. You can expect something like that coming up anytime. Sandeep Patil, thanks for you, ma'am. Oh, that's so sweet of you. Thank you, dear. Okay. Now write the differences between euchromatin and heterochromatin and name the virus used in Hershey and Chase experiment. Can you try this, guys? Name the virus used in Hershey and Chase experiment. Anyone who paid attention to the class, can you tell me? Hershey and Chase experiment, which virus was used? E. coli was used as the bacteria. There was someone to attack the bacteria, right? What was it? Come on, children, you can do it. You're superstars. No one? Keep on guessing. No. Hershey and Chase experiment, we just discussed it, no? There was a guy like this, which looked like spider. According to your NCRT, it looked like spider. It is what? The one that affects the bacteria, that is E. coli. It is... One more chance. Come on, Prachi, we can do it. Yes, it is bacteriophage. Okay, the name of the virus is bacteriophage. That is the one that affects. Now, what is the difference between euchromatin and heterochromatin? So, my dear children, our DNA is this much. Our nucleus is this much. Can we fit it inside the nucleus? Yes, with the help of some very important proteins which you call as histone proteins. So, there are 4 of them which comes like this. Okay, not 4 of them, sorry, 8 of them that comes like this. So over here you call this as octamer of histones, and onto this the DNA wraps around twice. How much? Twice. So there is a DNA that is wrapping around twice. Why does it wrap around twice? Because DNA is negatively charged, and the proteins which are there, they are made up of amino acids which are positively charged. What are the amino acids? Lysine and arginine. These are 2 positively charged amino acids. Okay. So now over here, since the coiling is happening, and suppose they are coiling up like this with no space, this is known as heterochromatin. Over here, this particular region is known as heterochromatin. Okay, fine. On the other hand, if you see this particular side, there are too much space in between the histone octamers that you see, which is why this is loosely packed, and you call this as what? Euchromatin region. So this is heterochromatin, and on the other hand, this would be what? Euchromatin. Now, why do you call it chromatin? First of all, so this particular structure, wherever histone proteins and the chromosomes comes to, and the DNA comes together like this whole thing is known as chromatin. This whole thing will be known as chromatin. When it condenses together more, it will be known as chromosomes. So over here, this is the heterochromatin and this is the euchromatin. Now, what are the features that you need to remember? Since they are very close to each other, it will be staining very darkly. So it is getting dark stained. On the other hand, this one, my dear children, would be staining lightly. Okay, this stains very much lightly. And one more very important thing that you need to remember, that is transcription will be happening in the euchromatin region. Why? Because you have some space for the RNA polymerase to come over here and do the transcription. But in the case of heterochromatin, this transcription will not be happening. Okay, so this is transcriptionally active, this is transcriptionally inactive. So in a typical nucleus, some loosely coiled regions of chromatin is there which get lightly stained and transcriptionally active, that is euchromatin. On the other hand, the chromatin that are more densely packed and stains dark and transcriptionally inactive is known as heterochromatin. Thank you, Sandeep. Thank you so much. Okay, I hope you got it, children. Now the last question I think of this particular chapter is this, right? They mention the criteria for a molecule that acts as genetic material. What all should be the criteria? First thing, it should be able to make the copy so that replication can happen, right? Otherwise, suppose it is not replicating my addition, do you think the copies can be transferred to the two cells which are made? No. And it is very much important that all the cells have the DNA, right? So replication should be there. It should be chemically and structurally stable. It should not be like, okay, sunrise is coming, this is coming, that is coming. It will change the structure, because if change the structure, the proteins will be mutated. Okay, and the gene will be mutated, and we itself won't be like this. So it should be chemically and structurally stable. It should provide the scope for slow changes. It's not like it should not show any changes. It should be there. Changes should be there, but very slow of them, such that evolution can happen. Good things can happen. If at all there is no changes, my dear children, do you think evolution will happen? No. So that is also a problem. So it should provide the scope for very slow changes. It should be able to express itself in the form of Mendelian character. That means it should be able to express itself. The gene should be able to code for some proteins. The protein should be responsible for all the characters out there. If it is not happening, do you think it's of any use? No. So these are the 4 things that you need to remember. These are the criteria for a molecule that acts as a genetic material. And which one is better, RNA or DNA? Children who are watching this video, can you tell me which one is better, RNA or DNA? Which one is better as a genetic material? Come on, come on, come on. DNA, exactly. Because DNA qualifies all of it. RNA is not structurally stable. Why? Because RNA has an OH group on this second carbon. We saw that pentose sugar is there. No, in RNA also this pentose sugar is there. But in RNA on the second carbon there is OH group, which makes it highly unstable. Okay? And in the case of DNA, this OH group is not there, which is why it makes it stable. Fine? Yes, M. Nauman. Nice name. Okay, can we go ahead with sexual reproduction in flowering plants? The 25 questions are there. I think since the time is going up very much, we will be restricting ourselves to solving MCQs. Okay, does it work? Are you okay with that? Shall we just solve the MCQs? If you're okay, give me a thumbs up. If you're not okay, then let me know. No, ma'am, we need to cover everything. So either way, you have to let me know. First question, MCQ, by then, okay, I'll try to solve, and by then you try to tell me the answer for the question that I asked. Do you need the MCQs or do you need the whole explanation as well? Yeah, MCQs is fine. Thank you, Ishak. Okay, select the correct sequence of anther wall layers. Okay, correct sequence of the anther wall layers, my dear children. In the anther walls, if you see, inside you have sporogenous tissue. Okay, this sporogenous tissue is something that is going to form the pollen grains later on. So which is why it needs nourishment. And who provides nourishment? It's the tapetum that provides nourishment. Always remember, the cells of the tapetum are binucleate. They have they have 2 nucleuses. Why? Because cytokinesis did not take place properly. Cell division did not take place properly, which is why they have 2 nucleus. And they have dense cytoplasm. Why? For nourishment. So this is the innermost wall, which is known as tapetum. The outermost wall is known as epidermis. Inside the epidermis, you have a wall that is known as endothecium. What do you call it? Endothecium. Inside the endothecium and in between the endothecium as well as the tapetum, you have one more layer which is known as middle layer. Okay, so I see a lot of you have given me the answer already. Let me see. Ishak says it's B. Okay. Sandeep also says it's B. Poma also says it's B. Prachi says it's B. So is it B? Let's see. Epidermis, then endothecium, then middle layer, and then tapetum. Completely correct. So E, and T. Okay, E, and T. Fine. So these are the wall layers, and you guys are absolutely correct. This is the picture that you would have to make in the sectional view of anther. Make it properly. Okay. Now, going ahead to the next question, let's see. Select the correct sequence of events in microsporogenesis. How is microsporogenesis happening? So already I told you the inside layer is known as sporogenous tissue. Inside the sporogenous tissue, each cells which is out there, they are known as microspore mother cell, or you also call it as pollen mother cell. Okay, so first thing is sporogenous tissue for sure. So I get two options like that. I can exclude all the other options. Now inside the sporogenous tissue, what is there? Microspore mother cell. Here what is there? Microspore tetrad. So this itself is wrong, but let's see anyways. So microspore mother cell, what happens to it, my dear children? What happens to it? So over here, okay, so one very important thing has changed. So those of you who have subscribed to the channel can only, their messages would be only popping up over here. So if at all you have not subscribed to the channel, please make sure you're subscribing. Okay, everyone says A, and B is also there. How is B there? It's A, right? So first of all, microspore mother cell will undergo meiosis 1 and you have two cells out there. Okay, this cell again undergoes meiosis 2 because meiosis happens in two stages. At the end, you have got 4 cells. What do you call these 4 cells as? You call them as microspore tetrad because 4 of them are there. Then they are going to move away from each other, and then finally, my dear children, you are going to get microspores, which is why your answer is A. So Prachi, Sandeep, Poma, as well as Ishak, all of you are doing great. You have said the correct answer. Now, the third question. Now I think you would be able to answer it better. The most resistant organic material present on the exine of the pollen grain is. So pollen grain which is there, my dear children, it has multiple layers. One is plasma membrane layer, then is intine, then is exine. And it is made up of the most resistant material out there. What is it? So can you tell me the answer in the form of 3A, B, C, D? That will be easy for me to understand. If at all you are telling the correct answer or not. The most resistant material is sporopollenin, my dear children. It can resist alkali, acid, everything out there. Which is why the pollen grains can be like acting like fossils because of this particular thing which is there. Okay, so all of you who told the answers, Sandeep, Poma, Prachi, Ishak, all of you, kudos to you, my dear children. And for those of you trying to answer and you're not able to, please subscribe to the channel. You will be able to do that then. Now, the functional megaspore in an angiosperm develops into option B. Option B, pectin is basically found in the intine, my dear, not exine. Intine is made up of pectin as well as cellulose. And that is not the most resistant material out there. The most resistant material is sporopollenin, which is found in the exine. Okay? Fine. The functional megaspore in an angiosperm develops into what? Children, can you tell me question number 4? Answer is this. That will be easy for me. I'll say you are correct, and you might be wrong. So which is why I don't want that confusion. So, in the case of megasporogenesis, the megaspore mother cell also undergoes meiosis. But you know what happens? 3 of the cells which are there, they're going to degenerate. And only one cell which is there, it will be there. And it is going to form the embryo sac. So, what do you call this type of development? Because where only one spore is going to develop. Anyone is able to answer? Where only one spore is going to develop, what do you call this type of development as? You call this as monosporic development. You call this as monosporic development, and your answer is B, embryo sac. Yes, Poma. Yes, Prachi. Yes, Ishak. Okay. Sandeep, also I think you gave the answer for this. Okay. Great. Fine. Let's go. Next, the following picture is an example of. Now let's see who all have read the NCERT really well. The following picture is an example of what? Children who have not subscribed to the channel yet, please subscribe. It's high time to do so. And who have not liked the video, click on the like button, like it right now. Who have not shared, please share it with your friends. So 5th question. This picture is an example of. So I see that a lot of you are opening your NCERT right now, which is why I do not see the fast answer. Multicarpally syncarpous condition is what you say. So basically over here, multiple carpels are there. This is 1, 2, 3, 4, 5, 6, 7, 8. All of them are there. And all of them basically are fused together. That means they just have a single ovary. But multiple, the upper style and stigma would be there, my dear children. This condition is known as syncarpous gynoecium. Where do you see it? Example. The name of this is example. What is this particular plant? It is Papaver somniferum. You might have studied in human health and diseases, opium poppy. Okay, so opium, you get it from this particular plant, that is papaver, and they have multicarpellary syncarpous condition. One more example is given. Hi Shamima. Hello, option B, you're absolutely correct. One more example is given in your NCERT. Multicarpellary apocarpous condition. Can you tell me the name of the plant? Everyone who is watching this video, can you tell me the name of the plant which has multicarpellary apocarpous condition? Multiple carpels are there, but all of them are separate. Any idea? Any guesses? Come on, come on, come on. No? So those of you who have subscribed can only answer, which is why I think you're not able to comment down. So please subscribe to the channel for answering. Again, you have opened your NCERT, guys. So no one, okay, I'm not going to tell the answer for that question. You have to find it and let me know right now. So if at all you're watching this video later also, you can let me know, but please let me know. Okay? Multicarpellary apocarpous gynoecium is seen in what? So multicarpous syncarpous is in papaver. They are fused, but there's a condition where they are not fused. Where do you see it? It's in your NCERT. Try to find it. Okay, next question number 6. Function of filiform apparatus is 2. What is the function of filiform apparatus, my dear children? Where do you find it? So this is the embryo sac. In the embryo sac you have 2 cells, synergids. Synergids are the best friends of your female gamete, which is the egg cell. So this synergids, my dear children, they have something that is known as filiform apparatus. Why do you think it is there? So that the pollen tube can be guided inside. They say the pollen tube to come inside, come inside. Here you have the egg cells. In the pollen tube, what do you have? You have 2 male gametes. So one male gamete is taken to the egg cell. Another male gamete which is there, basically it is fused with the nuclei out there, the polar nuclei out there. Okay? Got it? So filiform apparatus recognizes suitable pollen in the stigma, wrong. Stimulate division of genital cell, wrong. Produce nectar, wrong. Guide the entry of pollen tube. Always remember that. And when it does, my dear children, when the pollen tube goes through it, you know what happens? The synergids get killed. Okay, that is also there. So one friend has to sacrifice over here. So 6 answer is D. Ya Prachi. Yes, Sandeep. Sandeep. Yeah. No, Sandeep. And Poma, yes, it's D. Okay? It's not in the stigma, it's over here. Okay, in the embryo sac. Always remember. Let's go ahead. Clear? So now, from the statement given below, choose the option that is true for a typical gametophyte of a flowering plant. What is a female gametophyte? Female gametophyte means the one that will be having the gametes, that will be forming the gametes. And what is a female gametophyte, my dear children? It is known as the embryo sac. Embryo sac. Male gametophyte is what? Male gametophyte is pollen grains because it will be forming the male gametes. Inside the pollen grain, you have the male gametes. Similarly, over here, inside the female gamete, female gametophyte, you have the female gametes. So what is true about embryo sac? That is the question, and you have to tell me the answer. 7th question's answer. Any idea? It is 8 nucleate and 7 cell at maturity. It is free nuclear during development. It is situated inside the integument, but outside the nucleus, it has an egg apparatus situated at the chalazal end. Ma'am, I don't understand any bit of it. So let me tell you. Listen to me carefully, okay? So over here, from the 1 megaspore that you had, you're going to form the whole embryo sac. So the 1 megaspore which is there, it has 1 nuclei, right? This nuclei which is there, it is going to undergo 3 mitotic division. 3 times it is going to divide. First division, second division, third division. 3 mitotic division. Now how many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8. So 8 nucleate condition is true. 8 nucleus is there. Now, my dear children, how many cells are going to be formed of this 8 nucleus? Let's see that. So 3 cells are going to be arranged out there. They are known as antipodal cells. 2 cells over here, they are known as synergid cells. 1 cell over here, this is the female gamete, which is known as egg cell. And the 2 nuclei which is there, they are in this 1 cell. So 1 cell has 2 nuclei. Okay, so instead of having 8 cells, we have only 7 cells because 1 cell took the 2 nucleus. So 1, 2, 3, 4, 5, 6, and this is the 7th cell, which is known as central cell. And these 2 nuclei are known as 2 polar nuclei. So 8 nucleus, 7 cell condition is correct. Now the next option was it has free nuclear development. Free nuclear development means this only. The nucleus is dividing, but it is not covered by cell wall. That is known as free nuclear condition. Okay, so that is also correct. Fine. So these 2 options are correct. Where is it? These two things are correct. It is situated inside the integument but outside the nucleus. Is it true? No. So basically what happens, this is the ovary, inside that you have the ovule. Inside the ovule, my dear children, what do you have? You have the nucleus. So over here are the cells of the nucleus. Okay, and here is your embryo sac. And covering this particular ovule is what? Your integuments. So you have your integuments first, then the nucleus, and then the embryo sac. So it is situated inside the integument and inside the nucleus also, but here it's written outside the nucleus, so this is wrong. Now it is an egg apparatus situated at the chalazal end. So my dear children, what do you mean by egg apparatus? So over here, this thing that you see, the synergids and the egg, this is known as egg apparatus. And this side of the embryo sac, it is known as the micropylar region. Micropylar region, and this is the chalazal end. So always remember, everything important happens in the micropylar region only. Okay, micropylar region. So egg apparatus is in the micropylar region. So your answer would be what? Your answer would be A and B. A and B is what? C. So those of you said C, Sandeep, Prachi, Poma, as well as Sandeep, again, kudos to you, you guys are correct. Now those of you who are not telling me the answer, who are just playing the safe game, my dear children, better play the game over here rather than on the day of your examination. So if you want to prevent that, you can just tell your answers over here. Even though if it's not correct, I will make you correct. Okay, I'll help you with that. Fine. So I want to see children who are not worried about failures. I want to see children who are in the will to correct things out there. So which of the following is not a correct statement? Placenta is located outside the ovarian cavity. So for your understanding, I'll tell you, this is the ovary. This is the ovary, yes. And this cavity is the ovarian cavity, or you also call it as locule. And over here you have a thing that is known as placenta. Placenta is attached to the ovule with the help of a structure that is known as funicle. Okay, and the point of attachment is known as hilum. Now, placenta is located outside the ovarian cavity. Do you think it's outside the ovarian cavity? No, Baba, it's inside. So this is a wrong statement. First statement itself is wrong. Now, central cell develops into PEC after fertilization. The central cell, which had two nuclei, does it develop into primary endosperm cell after fertilization? Yes. So basically, the pollen grain carries two male gametes. One male gamete fuses with the egg cell to form the zygote. The second male gamete fuses with the two polar nuclei of the central cell to form the primary endosperm cell. Okay, so two things are being formed, primary endosperm cell and also the zygote. This primary endosperm cell is the one that nourishes the zygote to form it the embryo later on. Okay, so this statement is correct. Now, orchids contain more than one ovule in an ovary. This is given in an NCERT, and this is something that you never read. So yes, orchids definitely have more than one ovule in the ovary. They will be having, if more than one ovule is there, this ovule is something that forms the seed later on, so they will have multiple seeds also. Egg apparatus consists of one egg cell and two synergid cells. Yes. It is also correct. So what is wrong? It is A. So those of you who told the answer as A are correct, and none of you said that, which is why I am telling you, you need to tell the answers. You're not even trying. Then how will you get to know whether it's wrong or right? Answer is A. I think Sandeep tried. Good one, Sandeep. If at all you tried this question, good one. Okay, the answer is A. I hope it's clear to you. Please look at the structure. You will remember everything. Fine. Next question. Even in the absence of pollinating agents, seed setting is assured in. Even when pollinating agents are not there, still seed would be formed in what? Cucurbita, papaya, commelina, maize. Ma'am, where do you see that? You see in clistogamous flowers. Where do you see this happen? In clistogamous flowers, my dear children. Clistogamous flowers means closed flowers. Over here in closed flowers, the female part will be there as well as the male part will be there. Both of it would be there. Okay, so in this case, seed will be definitely formed. These flowers are known as clistogamous flowers, and you see it in Cucurbita, Oxalis and Viola. Cucurbita, Oxalis and Viola. So your answer, sorry, not Cucurbita, Commelina, Oxalis and Viola. So your answer would be what? C. Commelina, Oxalis and Viola. Okay, so your answer is C. Yes, Prachi. Prachi, I hope you gave the answer for question number 9 only. Children, are we here? Is anyone listening to me? So this is clistogamous flower, and the answer is C. They also have chasmogamous, the open flowers, which undergoes pollination. They need pollinating agents. Cool. The next question. Transfer of pollen grains from the anther to the stigma of another flower of the same plant is known as. So over here, this is a scenario. There is a flower over here, there is a flower over here. Transfer from here to here in the same plant. What do you call this? Autogamy, xenogamy, clistogamy, geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant is known as geitonogamy. Okay, this is geitonogamy. Yes, Prachi, you're correct. It's option number D. If at all it is happening between the same flower itself, the male and the female part is there, it is known as autogamy. If it is two different flowers crossing, you call this as X for xenogamy. Okay. Hello, Putta Raju Karthik. You were there the previous year is what I remember, until and unless I have another person with the same name. Okay, so 10th answer is what? D. Geitonogamy. Works? Chalo. Next question. In a fertilized embryo sac, the haploid, diploid and triploid structures are. In a fertilized embryo sac, the haploid, diploid and triploid structures are what, my dear children? Any guesses? So over here, the egg fuses with the male gamete. You get the zygote. So here N and N comes together. You get diploid, zygote out there. Next, the polar nuclei which is there, both of them has N plus N, right? Now one more male gamete comes together, again this also has N. Together all of it forms primary endosperm nucleus which has triploid content, okay, 3N. Now the antipodal cells which are there, they remain to be the same. The synergids which are there, they remain to be the same. All of them, my dear children, would be what? Haploid in nature. So in a fertilized embryo sac, the haploid, diploid and triploid structure are what? Synergids, are they haploid? Yes. Zygote, is it diploid? Yes. Primary endosperm nucleus, is it triploid? Yes. So what will be your answer? Your answer would be A. Yes. Sandeep, yes. Putta Raju Karthik, your eyes are correct. Your answer is what? A. Okay, other things are all wrong. Because synergids and antibodies, both of them are haploid. It cannot be over here. Cool? Next, in a monocot embryo, the shoot apex and the leaf primordia are enclosed in a hollow foliar structure that is known as. In a monocot embryo, this is the monocot embryo where the shoot apex, okay, shoot apex and the leaf primordia is enclosed in a structure. That means your plumule and the leaf primordia is enclosed in a structure. What do you call this structure as? Any guesses? It is known as coleoptile. So above the plumule, it has a P in it, it is coleoptile. Below the roots, it is coleorhiza, has R in it. You can remember like that. Okay, so Prachi, Puttaraju, both of you are correct. Your answer is what? B. Cool. Going ahead with the next question out there, which part of the fruit labelled in the given figure makes it a false fruit? I assume this is the last MCQ question that I have over here. Apart from that, there are 5-marker and 3-marker questions out there. So which part of the fruit labelled in the given figure makes it a false fruit, my dear children? C, D, B, A. What is this? This is apple. And what is a false fruit? When basically it's not the ovary which is alone going to make the fruit. It can be something else which helps in the formation of fruit. So usually the ovary becomes the fruit and the ovule becomes the seed. Suppose something else is also participating in the formation of fruit. This is known as false fruit. So over here, what is that part? The seed thing. Okay, the seed thing which is there, it is the thing that makes it false fruit, and it is thalamus. The thalamus is also contributing. Yes, Puttaraju Karthik. Yes, Prachi and M. Numan also tried to answer the previous question, I believe. Yes, so thalamus is this region where the ovary and everything is resting. Okay, and this forms the part of the fruit in the case of apple. So the correct answer is D. Done. Let's go ahead. So my dear children, I would leave these questions to you as of now, since the time we started at 5 o'clock. Okay, so the time is going up like that. So I would request you to go through it, and I have also explained it in the 2-marker, 3-marker question section in the midterm session series. Make sure you watch that video. It is going to help you really well. If at all you think you need my help, I am here. You can let me know in the comment section. You can put it down there. Okay, so this is the structure of megasporangium. I have explained everything. You have the PDF. I have explained properly in the videos. You can ask me, okay, if at all you have any confusion. So these are the 3 diagrams that you need to remember. One is of grass embryo, that means monocot, okay. One is a dicot. This is the structure of young anther, and there is a dicot embryo structure which also you have to study. Cool. These 3 diagrams can come. Characteristics of flower and wind pollinated flowers are very, very important thing. What are false fruit? What is parthenocarpy? Okay, the cells which are there in the pollen grains, one vegetative cell, the other one is generative cell. The vegetative cell has irregularly, the vegetative cell has irregularly shaped nucleus, whereas the generative cell has spindle shaped nucleus. You need to know that as well. Okay, where do we find the PDF? PDF you will find, if you join the WhatsApp channel, my dear children, you would be able to find the PDF out there. The link would be given there. Then the difference between gynoigamy and xenogamy could be asked. Please make sure you're going through that as well. Okay, and Sandeep said Malayalama. Nale class ida ma'am. Tomorrow the class is, math class would be there. Okay, now what is parthenocarpy? That question can be asked, where the ovary is developing into a fruit without fertilization. You find that in banana. Okay, so that's how you get the seedless varieties of fruits, my dear children. That thing, pericarp and perisperm. So the nucellus which is there, okay, outlining the embryo sac, my dear children, usually it is used up or degenerated, but sometimes it persists and it helps in the formation of the embryo. It helps in nourishment of the embryo. That is known as perisperm. Pericarp is the outer covering of the ovary, the fruit wall. Okay, that is pericarp. Ma'am, when do you complete the other chapters? So I am trying to complete human health and human reproduction now itself as an the 5 marker, 3 marker. I have already discussed in the 5 marker and 3 marker I have discussed in the midterm series, but if you want me to come live and discuss it, I can come, but you have to let me know in the comment section. You can ask me to come live once more in the comment section, and in that case possibly we can think of one thing like that. And my dear children, if at all you have not subscribed to the channel yet, subscribe to it. If at all you have not clicked on the like button, click on the like button right now. Okay, now we are going ahead with human health and diseases. Again, I'll discuss the MCQs. The 5 marker, 3 marker questions, I would request you to watch the midterm session video. If at all not, you want me to come live, as I told, put it down in the normal comment section of the YouTube that please come live. I will be definitely thinking of it. Okay, and I'll schedule a date and I'll let you know guys as well. Okay, the chemical structure is given below. Identify its plant source. What is this particular thing? Can you tell me the name of the plant? Okay. The chemical structure is given below. Identify its plant source. First of all, you have to know what is this particular thing. What is its structure of? This is the structure of morphine, my dear children. And where do you get morphine from? Morphine you get from opium poppy. And opium poppy is also known as what? Children, come on. Puttaraja, I did it from Indore, Madhya Pradesh. Okay. Sandeep, A. Sandeep says A. Atropa belladonna. No. It's not Atropa belladonna. Come on, children. Morphine, heroin, both of these drugs you get from Papaver somniferum, opium poppy. So you can remember like, oh, these are opioids. Morphine is an opioid. Heroin is an opioid. Both of them are drugs. So your answer is what? C. Okay. And this structure is of morphine. If you do acetylation of morphine, if you do some chemical changes out there, that's how you get heroin. Cool. Let's go ahead with the next question. Okay. One of the following is not the causal organism for ringworm. One of them does not cause the ringworm disease. It's a fungal disease, but since it's formed ring-like structure, you call this as a ringworm disease. So which is that particular thing that is not the causative organism for ringworm disease? Microsporum, trichophyton, epidermophytum or macrosporum? Which one does not cause? That is the question. Everyone who is listening to me, tell me the answer in this format. 2 A, B, C, D. And those of you who have not subscribed to the channel, let's subscribe. That's how you can put down the comments. Otherwise, you might not be allowed to do that because subscribers mode only is there. Okay? It's D. Puttaraja is saying D. Anyone to compete him and say it's not D? Sandeep also says it's D. Yes, it is D. So basically 3 types of fungi, okay, microsporum, epidermophyton, trichophyton, all of them are responsible for the ringworm disease. Whereas when you talk about macrosporum, it's not a type that causes the ringworm disease. So M. Noman, you're also correct dear, good going. Okay, so the answer is D. Now, an intestinal parasite which causes blockage of intestinal passage and whose eggs are excreted along with the feces of the infected person is. So there is a parasite that causes damage to the intestine. It causes blockage. It causes blood to move, it leads to loss of blood as well. And basically this parasite lays the eggs, and the eggs are released to the fecus of the human being. That means excreta of the human being. So what is this particular parasite? Is it Vicariaria bancrofti? Is it Ascaris? Is it epidermophyton? Is it microsporum? Your answer is, Puttaraja says A, but it is actually A or not. Any guesses? Is it A or not? A disease that you get when you do not have washed foods, when you have raw vegetables and things like that. You have to think like that. So excreta is going to the food and mixing it with the water, and that's how another person will get it. You get it? So it has to be transferred like that. It has to be disease like that. Vicariaria bancrofti is a worm that causes filariasis, and that is basically, so this particular thing, the worm is basically transmitted with the help of a bite of a mosquito, Culus mosquito. The larvae of it is transmitted like that. And that's how you get that disease. So it cannot be the one. So you have to rule that option out. What is the answer for this question? No one is telling me the answer. It is Ascaris, and it causes ascariasis. Okay, ascariasis, it causes intestinal blockage. Fine. The answer is B. Children, okay, I definitely understand that we have not prepared, but it's good time to prepare yourselves for the midterm examination. And if you have not, do not be sad about it, rather than work on it. Okay. So Ascaris causes ascariasis, and that causes intestinal blockage. Next question. Which of the following is affected by infection of Vicariaria bancrofti? Now you are asked about Vicariaria bancrofti. Which part of the body is affected because of this particular worm out there, my dear children? Have any idea? So those of you trying to comment down. If you have not subscribed, please subscribe. Then only you might be able to comment it down. Okay? Which of the following is not affected by the infection of Vicariaria bancrofti? So R.A. Tidagandi, okay. A. Sandeep Patil also says A. Yes, it is affected, sorry. So it is the lymphatic vessels which are getting affected. What does lymphatic vessels do? So you know, blood vessels, from your blood vessels, that is arteries, veins, they come together, and the junction is known as capillaries. They are very thin walled. From the capillaries, certain things move out, certain fluids like plasma, WBCs and things like that. So they go in between the cells. But do you think it's safe up there? No, because it can cause swelling. There should be someone to take up this fluid and return it back to the blood. What is that? It is the lymphatic vessels. Okay, it is the lymphatic vessels. Now this particular the worm out there, okay, it basically affects your lymphatic vessels. It blocks your lymphatic vessels. Now do you think that the fluid can be taken up? No. Which is why there will be swelling. There will be deformities in your genital area. Wherever your lymphatic vessels are there, for example, your leg. Mostly it is a leg that is being affected. So that is a thing. Okay, so your answer is A. Mood baby, definitely giving you the PDF once the lecture is over. Okay. So Sandeep as well as Puttaraju as well as R.A. Tidagandi, you guys are correct. Okay, chalo. Next question. Which of the following pairs correctly matches a disease and a pathogen causing it? Matches a disease and a pathogen that is causing it. So typhoid, is it caused by Salmonella typhi? Is it caused by Salmonella typhi? A. Answer is A, B, C, D. What is it? So R.A. Tidagandi says it's A. Puttaraju says it's A. Okay, it's actually A. Typhoid is caused by Salmonella typhi, which is a bacteria. Pneumonia is caused by Haemophilus influenzae, not Haemophilus pneumoniae. So don't get it wrong. Malaria is caused by Ascaris lumbricoides. It's wrong. It is caused by Plasmodium. Ringworm is caused by microphyton, microsporum, epidermophyton, trichophyton, all those things. But not Entamoeba histolytica. Entamoeba histolytica causes what? It causes amoebic dysentery. Okay, so your answer is A. Mood baby, definitely I'll be sending you the PDF for human reproduction once. Okay, don't worry about that today itself, once the class ends. So my dear children, the answer is A. Now for those of you who are watching this video and not replying and not giving any answers, it's a very sad thing to see. Why Baba? Why are you not replying? Try to find the answers by yourselves and let me know. However wrong it is, it's totally okay. It's not the end of the world, okay? So hemozoin is what? Hemozoin is a precursor of hemoglobin. Does it make hemoglobin? No. Toxin released from streptococcus infected cells? No. Toxin released from plasmodium infected cells? Yes. Why do I say so? Why do I say so? Because my dear children, plasmodium is the one that causes malaria, and plasmodium, they feed on our RBC. You know, in our RBC, they take the hemoglobin and they break down the hemoglobin into heme and globin. This heme which is there, it is very toxic for them, which is why they converted it to something that is known as hemozoin, so that it becomes less toxic for them. But you know what? It becomes less toxic for them, but when the RBC is bursted and this hemozoin is released, it becomes toxic for us, and that is why we had fever, chills and everything out there. Okay, so your answer for this question would be C. Cool? So this is a whole chart that I have made already. You can refer to it. It can be really, really helpful for you. Now going ahead with the next thing. During the life cycle of plasmodium, gametocytes develop in. Where do the gametocytes develop? So over here, if you see. So basically, first of all, the plasmodium enters in sporozoite stage. Okay, from the saliva of the mosquito, it enters in the sporozoite stage. It gets into the human body, and the first place it goes is the liver, okay, of the human body, and it affects the hepatocytes, that is the liver cells, and there it undergoes the asexual reproduction. It increases its number, and the hepatocytes, they get lysed. And when they get lysed, what happens, my dear children? All this plasmodium things which are there, they go to the blood. Now in the blood, they form the second cap inside the RBC. Inside the RBC, again they will undergo asexual. There is no sexual mode of reproduction happening in the human body. It's always asexual mode of reproduction. It happens over here, and again when asexual reproduction happens, this particular RBC gets burst open, and my dear children, hemozoin will get released, which is why we get fevers and chills. Apart from that, they will now form a new, a new stage of it, which is known as gametocyte stage. Gametocyte stage is the one that will later lead to formation of gametes, but they don't form gametes here in our body. So these gametocytes are formed, and now it is released in the blood. This blood is taken by the mosquito, and in the mosquito's gut, the gametocytes will be forming the gametes out there, and the gametes will be fusing in the mosquito's body. So the gametocytes are formed where? In RBC itself. In the RBC of human being. So you have to write the answer like that. So during the life cycle of plasmodium, gametocytes develop in what? Human RBCs, or you also call this as human erythrocytes. Mood baby is asking tomorrow which class is there. It is mathematics, my dear children. Okay. Now the next question. Okay. This is not human liver, okay. This is human RBC. The human immunodeficiency virus primarily attacks and destroys what? The human immunodeficiency virus primarily attacks and destroys what? RBCs, T helper cells, platelets, T cytotoxic cells. So first thing is that HIV viruses go into the macrophages and increases their number, which is why macrophage is known as the HIV factory. But once they move out of the macrophage, they first go and affect the T helper lymphocytes. Always remember, helper ones are the ones that is affected. Yes, Kumda, you are right. And even R.A. Tilagunde, you are also correct, my dear child. Puttaraju is also correct. It is B. T helper cells are the one which are getting affected because they are the one that helps in immunity. If they go down, everything else will go down. Okay? So always remember that. Next question. The substance produced by a cell in viral infection that can protect other cells from further infection is. So cell is there. Suppose it is infected by some virus. Now this cell, to save the other cell from virus, what will it release? It will release something that is known as interferon. What does it release? It releases interferons. So your answer for this question would be C. Kumada, totally active. Okay, Sandeep says it's B. Dear, please check the answers. It is interferon. They are going to interfere and say that, hey cell, you have to be safe from all the viruses out there. Okay, so your answer is C. Yes, Sandeep. Now you're correct. Next question. Antibodies present in colostrum which protect the newborn from certain diseases is? Yes, Puttaraju, it's cytokine barrier. Cytokine means a cell releases certain secretion that is known as cytokine. So interferon is a type of cytokine. So it comes under cytokine barriers. You're correct. So antibodies found in colostrum. What is colostrum? The breast milk, the first milk of the mother, which is yellowish in color. It is given to the newborn. Why? Because it is ready-made antibodies that you are basically passing. And what is it? It is IgA. You're correct. Who said A? B, B, B? B, B. Puttaraju, Karthik, Sandeep said B. And okay. Mood babies, 8th question. Okay, now I get it. So we're talking about the 10th question, my dear children, and it's IgA. You can remember A for Amma. Okay, so from the mother you get IgA. Cool. I hope you'll never forget it. And my dear children who have not subscribed to the channel yet, please make sure you're subscribing and share it with your friends also who are in need, and do not forget to hit on the like button if you're liking the video. Now, vaccines work by, how does vaccine work? By stimulating the production of antibodies, by killing the pathogens directly, reducing inflammation, blocking the entry of pathogens into the body. How does vaccines work? So in the COVID time, we were given vaccines. Why? Because these vaccines have the ability to ignite the immune response in us. They're not going to cause trouble in us. They're going to ignite the immune response such that the body remembers, okay, this has come. Now the next time an active thing comes in, my body will react very well to it. So it is stimulating the production of antibodies. So which is why when the first time I got the vaccine, I got fever because my body was reacting to this. So it is usually weakened pathogens or attenuated things, okay? So weakened pathogens or some part of the pathogen which cannot cause any trouble in our body, that is being taken. Okay, so your answer for this question would be A, stimulating the production of antibodies. Cool? Done? Yes, R8, Idagodi, Kumuda, all of you, my dear children, are doing absolutely correct. Mood baby is also correct. Okay, next question. Which of the following cells actively participate during allergy? During allergy, which is a cell that actively participates? Is it B lymphocytes, liver cells, mast cells, red blood cells? What is it? It would be... Chalo, I'll wait for you guys to answer. B lymphocytes, liver cells, mast cells, red blood cells. 12B, liver cells are. Think. R8 Idagodi said it's C. Yes, it is C. So basically there are some cells which are known as mast cells, and mast cells on their top you have some antibodies, that is IgE, allergy, IgE. Now basically on this antibodies, if at all allergen comes and attaches, my dear children, this mast cell which is there, it becomes active and releases two things. One is histamine and the other one which is there, it is known as serotonin. Histamine and serotonin, both of them are responsible for one very important thing that is known as vasodilation. The blood vessels are going to get dilated. So many other cells are going to come over here and say that something is happening, we need to do something about it. That's why you get inflammation, you get runny eyes, lot of things. Okay, so mast cells or must cells, you can remember like that. Okay, they're helping in allergy. And IgE also, please remember that. Now, my dear children, 5-mark question. As I said, I would not be discussing it as of now because of the time constraint. But I had explained it in the midterm session series. In every chapter that I have done, I have given MCQ. Sorry, I have given one-shot video and then all the questions together. So you will find the solutions for all of it. I mean, explanation for all of it. But if you want me to come live again and let you know the explanation, I can do that. Put it down in the comment section. I'll definitely do that. This part is not there for your midterm. Okay, so primary lymphoid organs, you have to talk about them. Primary lymphoid organs are bone marrow and thymus, where the cells are being formed first. They become antigen sensitive, but they don't become effective. They become effective when they go to secondary lymphoid organs such as spleen and lymph nodes and things like that. So you need to explain. Where do we need to find one-shot video? It's there in the midterm session series. If you find, go to the playlist of Diksha Karnataka, you will find the midterm session, and in that biology will be there. For all the chapters you will find in the one shot. Okay. Now explain different techniques in which cancer detection. So basically talk about biopsy. CT scan is there where you use X-rays, and X-ray, basically CT scan is for finding the internals of an object. MRI is comparatively safer because you're using non-ionizing radiations, my dear children, and it is for finding problems in the living tissues. Then antibodies against cancer-specific antigens that are also being taken, but all of it we have discussed previously. These are certain drugs. So sedative and painkiller, it is morphine. Something that is used for treating insomnia if you're not able to sleep, and also depression, it is barbiturates, amphetamines, benzodiazepines. Blood pressure, always remember, blood pressure is linked to smoking. Okay, in the smoking, tobacco has nicotine, and nicotine increases the blood pressure. Okay, and depressant is heroin. Heroin makes it. Have you watched the movie Heroin in Hindi? In that, the lady is in depression, so you can remember like that. Okay, affects the cardiovascular system. There is only one thing that affects the cardiovascular system, and that is cannabinoids, cannabis sativa. Okay, the thing that you get from the cannabis sativa, such as hashish, marijuana, and all of this thing, they affect the cardiovascular system. Rest of it, most of it, they affect the CNS, central nervous system. Okay, but this is cardiovascular system. The schematic representation of replication of viruses, you have to talk about everything that is happening in this particular picture. Make sure you're watching the video, my dear children. Over here, 3 properties of cancerous cells. I have explained it there. Active and passive immunity, difference between that. Okay. PDF, we are not uploading in the YouTube, rather in the WhatsApp channel to make it more segregated for everyone, for you to get it, access it later on also. Otherwise, you would have to search here and there, right? Okay. Type of carcinogens, physical, chemical, biological. Cancer as a whole is very important. This picture of antibodies is important. Make sure you're going through it. Okay. And HIV transmission, how does it happen? Everything. Now comes to the last chapter of today, that is human reproduction. We will see the MCQs and then we'll stop the video. We'll not go any further. Okay. And mood baby, as I promised you, I will be giving you the PDF. Don't worry. Today itself. So scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. So how much degree lower than the normal body temperature? 2 to 2.5 degrees Celsius lower than the normal body temperature, which is why scrotum is outside. Okay, so that testis remains in that temperature. Otherwise, it won't be able to form the sperms. Now, each testis has about B compartments. How many compartments are there? How many testicular lobules are there, my dear children? How many testicular lobules are there? 250. Okay, and now measure C centimeter in length. What is the length of the testis? Always remember the width is 2 and 3 and the length is 4 to 5. So your answer is this, B. Madhu Priya HD, ma'am, do KCET chapters. These are also going to come in KCET. Okay, so these chapters in the PU2 obviously are going to come in your KCET examination also. So make sure you're covering all of it nicely. We will be specifically focusing doing on KCET, and for those of you who are looking for a coaching, there is KC90, our own thing. Make sure you're going through it. I have included in the video. The link will be given in the description also. You can check the website for getting more details about KC90. Okay. Now, during embryonic development, embryo's heart is formed at the end of. When does heart form? At the end of first month. Okay, so the first thing that we check when we die is heartbeat. Similarly, the first thing that we check when someone is alive is the heartbeat. So heart is formed first, and after the first month only you can find it. So answer is first month. Here is the table of all the things that happens in all throughout the month. Make sure you're reading it properly, not missing out on anything out there. Okay? Fine. And this is 5th month, not 6th month. Cool. Next. The vas deferens receives duct from the seminal vesicle and opens into urethra as. So vas deferens, my dear children, comes like this, and it receives a duct from seminal vesicle. Both of them, my dear children, it would be opening together by forming the ejaculatory duct, and then later it opens into what? The urethra out there. So the thing is ejaculatory duct. So I have here given you the picture. See, this is the vas deferens that comes. This is the seminal vesicle. Both of them, they open into ejaculatory duct, and ejaculatory duct basically opens into the urethra. Okay, so your answer for this question would be B. Cool. Now, the function of myometrium layer present in the uterine wall is to. the myometrium. So 3 layers are there. Perimetrium is the outside layer, myometrium is the middle layer, and inner layer is endometrium. Myometrium is made up of smooth muscles, and smooth muscles are there for what? Contraction. So R.E. Tidagodi says it's B. Is it B? Yes. It is made up of smooth muscles and it is there for contraction. So I'll say exhibit strong uterine contraction during the process of parturition. That means when the baby is about to move out. So that's when we need it most. Okay? Not undergo cyclic changes. Cyclic changes are undergone by the innermost layer, which is endometrium. Cool? Works, give protection to the uterus, that is perimetrium. Helps in implantation process, again it's the innermost layer, that is endometrium. Okay. Sertoli cells are found in. Where do you find Sertoli cells? Kumuda, Mood Baby, Puttaraju, R.E. Tidagodi, you guys are doing great. Okay, I am seeing all these comments. Okay, Sertoli cells are found in where, my dear children? So this is seminiferous tubules. Inside the seminiferous tubules, you have Sertoli cells and also the germ cells. The germ cells are the one that later forms the gametes. Sertoli cells provide nourishment for this. This is why we call them nurse cells as well. So where is it all happening? Inside the seminiferous tubules. So Sertoli cells are found in ovaries and secrete array. No. Adrenal cortex, no. Seminiferous tubules and provide nutrition to the germ cells. Yes, ma'am. Okay. So answer for this question is what? C. Yes, Kumada. Yes, Puttaraju, Karthik. You guys are correct. Going great. Now the cells that secrete testicular hormones, androgens are. The male sex hormones are known as androgens. Testosterone is one among them. Okay, what are the cells which secrete it? So over here I told you seminiferous tubules is there. In between the seminiferous tubules you have gap, interstitial space. And here you have what? The next type of cell which is known as Leydig cells. Okay, Leydig cells. You also call this as interstitial cells which release the androgens. Okay, so your answer would be B. Kumuda as well as Puttaraja has got it correct. Those of you didn't get it correct, check answers once, okay? Now, seminal plasma in human males is rich in. Seminal plasma means secretion of all the accessory glands together. Accessory glands like seminal vesicle, prostate gland and bulbourethra. Seminal vesicle releases fructose so that it provides energy to the sperm. Prostate provides calcium as well as enzymes. Okay, which is very important for the process of fertilization. And bulbourethral is there for lubrication. Remember, both these glands are paired. This is unpaired. Prostate is unpaired. It has P, but it is unpaired. So, seminal plasma, the secretion of all of it together is rich in what? Fructose and calcium? Yes. Glucose is not there. DNA is not there. Ribose is not there. So, your only answer is what? A. B. Okay, I see that answers are coming a bit slow out there, but it's okay. Okay, it's A, okay? Question number 8, answer is A. Now, next question. The process of release of sperms from the seminiferous tubules is known as spermatogenesis? No. The process of release of sperm from the seminiferous tubules is known as spermiation. This is not spermatogenesis, it should be spermiation. So, this statement is wrong. Aviation, you can remember like that. It's release of sperms from the seminiferous tubules, right? The spermatids are transformed into spermatozoa with the process that is known as spermiogenesis. Is that true? So at the end of meiosis, you get something that is known as spermatids. They are immature. And to make it mature, we convert it into spermatozoa, which looks like sperm. This process is known as spermiogenesis. That is correct. That is correct. Statement 1 is false. Statement 2 is correct. Statement 1 is false, but statement 2 is correct. Yes, you guys are correct. Kumuda as well as Puttaraju. The answer is D. Okay, the answer is D. Trishank is here saying the answer is D. You are absolutely correct. R.E. Tidagodi is solving the 7th question. Yes, I'm a bit ahead, so I am not sure. But thank you for trying. Now, question number 8, this is question number 10, which is very, very important. In spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is affected? If FSH hormone is not secreted from the anterior pituitary gland, which stage is affected, my dear children? What do you think? So, FSH which is there, it goes and acts on the Sertoli cells. And the Sertoli cells in turn are going to produce androgen binding proteins, or you can just remember some factors. Where do you get the androgen from? From the Leydig cells. So LH, which is another hormone, acts on the Leydig cells, and that's how you get the androgens. And androgens comes over here for the process of spermatogenesis to happen smoothly. And at the end of spermatogenesis, I already told you there is spermiogenesis, where spermatids are converted to spermatozoa. This needs high amount of testosterone, and for that, for testosterone to stay there, you need some factors, and that is released by the Sertoli cells. So if at all FSH is not there, Sertoli cells would not be producing these factors, and hence spermiogenesis would be majorly affected. Okay, spermiogenesis would be majorly affected. So now you tell me, formation of primary spermatocyte? No, formation of spermatozoa, because at the end of spermiogenesis you get spermatozoa. So this process is affected. Okay, cool. So your answer is B. No one gave me the answer for the 10th question, is it? So my dear children, those of you who didn't understand, spermatid to spermatozoa conversion is known as spermiogenesis, and that needs these factors. So if this conversion is not happening, obviously this is not going to be formed. Cool? Fine. Now, to produce 2000 sperms and 400 ova, how many spermatogonia and oogonia are needed? Just a second, okay? How many spermatogonia and oogonia are needed? 2000 sperms. So, in one particular spermatogonia produces 4. You can remember like that, 4 sperms. So, if 2000 sperms are there, then 2000 divided by 4, how many will you need? 500. Similarly, 400 oogonia. One oogonia releases, basically at the end you get 1 egg only. 1 egg. So, if 400 ova is there, then 400 divided by 1 is just 400. So, 400 oogonia and 2000 divided by 4, 500 sperms are going to be produced over here. So, your answer would be this. So, the 500 spermatogonia need to be produced. Okay, so your answer is B. Yes, Puttaraju, as well as Kumuda, as well as Trishank, you got the answers correct. So, please be remembering about the number of sperms as well as the egg. Cool. Next question. Which among the following has 23 chromosomes? Spermatogonia, zygote, secondary oocyte, oogonia. 23 chromosomes means half the number of chromosomes. Yes, Puttaraju. I got it. You were correct. You told the answer first. Good going. Which among the following has 23 chromosomes, half the number of chromosomes, my dear children? Someone that has underwent meiosis. Spermatogonia, it has not underwent meiosis yet. It will undergo later on. Zygote is a fusion of both of things. So no, for both of the gametes, so it is diploid in nature. It will be having 46 number of chromosomes. Secondary oocyte, it has underwent meiosis, which is why it will be having half the number of chromosomes, that is 23. Oogonia has not yet started having division, so which is why again it is not. So your answer is C. So Puttaraju, Kumuda. Okay, so over here I think Kumuda answers first, and then Puttaraju, then Trishank, then again. Okay, great going children. I see the energy out there. Now, identify the incorrect pair with respect to menstrual cycle. What is incorrect about menstrual cycle over here? Formation of corpus luteum 15 to 28 day. Does it happen like that? Yes. 15 to 28 day corpus luteum is being formed. Secretion of progesterone 15 to 28. Corpus luteum is the one that secretes progesterone, so that is also correct. The regeneration of endometrium 5th to 13th day. Endometrium, okay, from 1st to 5th day, endometrium falls off. From 5th to 13th day, it slowly regenerates. So that is also correct. Disintegration of endometrium 5th to 13th day. No. 1 to 5th day, endometrium is disintegrated. Later, 5th to the 13th day, it is being formed. Then it is formed better. Okay. So your answer over here would be what? D. Puttaraju, yes, you're correct over here. Incorrect pair with respect to menstrual cycle is D, my dear children. Okay. This is known as follicular phase or also proliferative phase, where it actually proliferates, does not disintegrate. Now, which of the following statement is true? LH surge induces menstruation. LH surge induces ovulation. The Graafian follicle ruptures and the secondary oocyte is being released. It does not induce menstruation. Okay, please remember, it does not induces menstruation. This is false. Regression of corpus luteum increases progesterone. That means when corpus luteum dies off, do you think progesterone increases? No. Corpus luteum is the one that releases progesterone. So this is again wrong. Diploid egg is formed after second meiotic division. Diploid egg is formed after second. Diploid egg concept is not there. Especially after meiosis, you get haploid egg, no? So this is also wrong. Embryo differentiates from inner cell mass of the blastocyst. Yes. So there is blastocyst stage where outer cells are known as trophoblast, and there is inner cell mass which forms the embryo. So your only answer becomes D. Okay? Okay, I have Bhiru also over here. Someone new out there. So let me see who said Puttaraju first, and then Trishang, and then Bhiru, and okay, Kumuda also doing great, my dear children. Okay. So match the items given in column 1 with those in column 2 and select the correct option given below. So I think, assuming that this is the last question, match the items in column 1 with column 2. So proliferative phase is also known as what? Follicular phase. Okay, proliferative phase is also known as follicular phase because follicles are being generating out there. Secretory phase is also known as luteal phase because corpus luteum makes progesterone, and progesterone makes the endometrium secretes like glycogen. Okay, and over here, menstruation is also known as breakdown of endometrium. So C goes with 1, and if you know that, I think half of your problem is solved. This we can exclude. Okay, and A goes with 2, A goes with 2. So this is your answer. Let me see who all got it correct. C. Okay, Kumuda said C. Nagaraj is back. Kaputtaraju also says C. Guys, you're going great. Okay. Now, the secondary oocyte when released is covered by a non-cellular layer. What do you call this? Secondary oocyte makes its own layer, and what is it known as? It is non-cellular. It does not have any cells in it. It is known as the zona pellucida layer. Not plasma membrane, not corona radiata, not cortical layer. It is zona pellucida. Okay. And my dear children, with that, I think we are completing the MCQ question. As I said, because of the time constraint, it's too much late. Some people are waiting out there for some things. So I would stop the live up over here. And I would request you to watch the midterm playlist if at all you feel that, okay, you need some understanding of any concept. I have made one-shot videos. I have given you questions videos separately. Okay, again in one shot itself. So you can watch it. And if you need my help, if you need me to come live once more, I can do that. Just let me know that. Okay, I'll just quickly show you the important things. This picture is important. A sectional view of human female reproductive system and mammary gland. Please try to draw it. And also the sectional view of male reproductive system. Sectional view is actually this, but they mean this. Okay, so please practice this particular thing as well. And now spermatogenesis, the schematic representation of how it works, all of it is very important. Events that occurs in the ovary and the uterus during the follicular phase. Each phase, whatever happens, menstrual cycle as a whole is very important. Parturition is induced by neuroendocrine mechanism. That means neurons as well as hormones are equally responsible. That is what you mean by this. So how does it work? I have written over here. Again, I have explained in the series. Please watch it. What are the organs that releases these hormones in menstruation? There are two things. One is pituitary gland, other one is ovary. You need to know that what hormones releases what. Okay, and in pregnancy, certain hormones increases, the placental hormones, HCG, human placental lactogen, estrogen, progesterone, all of it. Okay, so please that as well. And that's all, my dear children. If at all you have not subscribed to the channel yet, make sure you're subscribing, and notes I'll give you right now after I stop it. And if at all you think that this video was helpful for you in any ways, not forget to share it with your friends. And as I said, do not forget to let me know in the comment section if you have any doubts. If you feel this video was helpful for you in some ways, let me know that as well. Do not forget to hit on the like button. And wishing you all the best, my dear children. If you want a live session again, put it down in the comment section. We'll think for it. Okay? Definitely doing that anytime. And that's all. I'll give you the notes right now. Do not worry about any of it. Bye-bye. Lots of love. Please take care of yourselves. Bye-bye. Okay? I'm assuming everything is done. Shall I stop the live? Bye-bye. Trishang, bye-bye Puttaraju Karthik. Okay.